was up all night helping map this brand new Seamount.
No.
Oh no! I'm going to give you all of these kudos. It's going to be like a
how
big
about
box of
Kevin and
granola
Nia
bars.
and Meshcore? But we basically what happened is Kevin and Nia started the mapping last night with Charlie, and we all got up around 1:30. Meshcore came and woke me up, and we went and took a look at what was going on at that point at 1:30 and watched for about half an hour till we understood what was happening. Which seamount, we were actually going over 3 seamounts, which looked like it was the best target, and had a plan. And then I went to bed for a couple hours and got up at 4, and we regrouped, and everybody came in. Chris Ritter came in, the ROV acting lead right now, and then we talked about the actual dive site and figured that out. So it's really Kevin and Nia and Mashcore that really put in a lot of effort with Charlie as well. Excuse me, with Charlie as well, late in the hours, all night last night, as they have every night.
Other way, yeah.
So
that's right, and that's a side of the Okeanos Explorer crew that is often not immediately evident from our broadcast.
All right, so it's cheating
and
to
that's
your starboard.
we
So
have
let's rotate
a
backport.
dutiful crew of people who
it'll
map
stay there.
the seafloor during the night shift,
just
composed of
stay off
the individuals that Chris just mentioned, and they stay up all night, essentially monitoring the devices and the equipment that give us the detail that we need in order to
go to 4,
descend
5.
to the bottom
We'll just
and
descend at 4, 5.
move around without any kind of harm.
yeah, so we haven't really seen a complete compilation of the data from last night. We saw the one swath that they had mapped on our route over, but I'm counting right now possibly part or all of 8 seamounts that have never been mapped before have been mapped by this group, Mashcore's group at night.
I think that's good.
In the monument, and of those, at least 6
Command
of
Honor
those
deputies
are
coming
almost
down,
complete or completely mapped.
Just keep
So
an eye
it's
on
been a very, very productive mapping cruise, even though our primary function here is the ROV dives. The mapping always supports, and so we've got a lot going on here, and I just saw all of the seamounts from Nea. She gave me copies of them. I put in ArcGIS, and they're just beautiful. It's beautiful data.
Yeah, I expect that you'll be acquiring bottoms soon. I'm going to switch rotation center to A-frame.
2 enter is tight.
Sediment bottom. I think Bruce Mundy got his wish.
You so happy?
And so Scott France also was quite interested in seeing something a little different.
Bail out 215.
Just let it sink for a second.
So did we?
I was hoping that the channel here wouldn't be sedimented, or at least not thickly sedimented, and that might be an indication that the currents moving through this compressed region would be scouring the bottom. So I think I see there's ripples, so the current is strong enough to create ripples, but not strong enough to wash away all sediment.
All right, thank you. And that was the voice of Scott France from the University of Louisiana at Lafayette.
They're coming now.
Everybody who's been tuning in for the last few years should be very familiar with his voice. He's been an absolute tremendous authority and resource for the Okeanos cruises, both in the Atlantic and in the Pacific. And he knows a lot about a lot. That's all I can say.
So the title of the expedition is La Lima o Kamawana, which essentially means partnership in the ocean, or partnership for the ocean. And one of the aspects of that, which is reflected in our science team, we have a team of experts which we are able to communicate with instantaneously on shore. These include experts in corals of many different types. We have geologists, ichthyologists, people who study fishes, and these people are able to communicate with us instantaneously via telepresence. So that, in cooperation with our pilots and with our mapping crew and with the many individuals who operate the ship, the Okeanos Explorer, that is cooperation is essential to the endeavors that we're engaging in here,
Yeah,
exploring this never before explored region.
Yeah, so to update those of you who aren't on the call this morning, the dive plan is actually to drop down into a little bit of a saddle. It almost looks like a crater or a channel on the very top of the seamount, and that's what you're seeing. We were expecting to see soft bottom based on the backscatter, and this is a nice, good landing site, and also to accommodate, Bruce Mundy, who thinks he's going to see more fish in the sediment area. He didn't get any yesterday.
How's
But
tether look?
then we are going to move over toward a ridge-like summit
down a little bit,
topography feature,
not if it
and we're going to go up the slope
the
of
tether
this.
looks
You'll
like
see
it
a
might come too
conversion
high
or
up on the
transformation from sediment to rock. We'll get up a little into a little saddle area, get on top of the crest, and then we'll run the crest up. And it's an upslope,
when you're
so
happy
even
with
though we're
that.
on the crest of kind of a ridge-like feature, but not really, we'll continue up along it. So it'll be upslope most of the day on rocky habitat, but dropping in the sediment gives us a look at the saddle area. And just for your information, those of you watching, we are facing about 90 degrees orientation to the channel itself, so those bed forms indicate that the water is moving through this channel that we've dropped into from north to south. Got little cobbles. I wouldn't call these necessarily manganese nodules, which usually form beds on sedimented abyssal seafloors.
It's almost kind of a talus.
Yeah,
That maybe you
very
wanna-
scattered if they are
We should either get
nodules,
a
but I
move
don't
in,
think so. They're different
or you
sizes.
should scoot back
Yeah,
a
I
little
think
bit.
they're just manganese coated
I just think you're pretty far out there in
Pebbles
front of me.
pebbles.
Cobbles
we were
and
just
things.
doing tether
Thank you.
maintenance in
Oh, okay,
the
gotcha.
last 10 minutes pretty much.
Okay,
It took us a while to come down.
all right. I'm gonna come down a bit.
Yeah, just keep an eye on
So you may have to
Yeah, see,
complete
those
a lot
are
of my
mostly
sentences this
Yeah, it's
morning,
kind of
Chris.
blowing up
That's
slope
fine.
here.
Yeah, we actually
I'm
turned
always happy
east
to.
Yeah,
I'm always
and then
happy to
turn
speak for other
back.
people, Chris.
yeah.
to try
I'll
to
just
get us
actually I can
to
just
the
pretend to be you.
Okay.
People are probably confusing our names at this point. This is Chris Kelly
It's always fun when the tether's blowing up in front of you.
from
But it
the
looks all right.
Hawaiian Undersea Research Laboratory.
Yeah,
Say it again? Sorry.
Go ahead. It looks all right.
Oh, yeah.
Yeah.
I'm trying to see what our really
Yeah,
bad
thanks.
Oh,
joke
get your swing
assortment
arms
looks
out.
like
Yeah,
for
we have
today.
swing
I
arms.
actually
And,
listed some on my
As soon as we get a
iPad.
swap here, yeah, we
I'll
should
start you out if you don't have
get
one.
moving. Doesn't look like there's a whole lot to see.
Okay, you're
Yeah.
on, Chris.
It is.
Beware.
Sandy.
So this one comes from a member of our onshore community sent via Twitter. What do you call a sea cucumber that is just eviscerated?
Oh boy.
You mean like expelled his innards?
That's right. So sea cucumbers can expel their innards as a defensive mechanism.
So again, what do you call a sea cucumber that just eviscerated?
That has just thrown its guts out at you.
I don't know. What?
You call it a holothurian.
Oh,
Auto-hitting,
Science
man.
joke, I guess.
You're looking at
Do you
generally
know
like a
who sent that in?
050,
That was quite good.
That was
060.
Megan McCullen.
Yeah, I think,
it was sent to me via
someone
Twitter,
had
but
back
she is
with
part
C.
of our
060 is what I got.
onshore
Okay.
I'll
science
select
team.
2.2 for you, sorry.
And I
Okay.
believe
When
she
that
gave us
direction
some
looks
assistance
pretty
good. Well,
with
you got
some
it.
bryozoans
It's
yesterday.
Oh,
Chris,
Megan.
wouldn't that be a holothurian?
You want to kind of back up a bit until we get the ship moving?
Yeah, sure.
Just because when
Your
we're
turn.
stretched out, it makes that tether.
Megan, do you really want to
Yeah.
enter this
Well,
world
this
with
is
Chris
serious.
and I and this quality of humor? I don't know, but that was very good from our perspective. Thank you, Megan, for doing that.
Yeah, it's looking better.
So taking a look, we've got Asako Matsumoto from the Chiba Institute in
Okay.
Japan, Jessica
I'll, keep
Robinson,
your lights
Bruce
out.
Mundy from the NOAA Inoue Regional Center on Ford Island in Hawaii. We've got some OER folks, Emily Crum. We also got URI, University of Rhode Island, ISC. It's
Yeah, Nav,
the
whenever that around
crying out loud. I'm going to have a
What's that?
bad morning. Forgot center, the inner space center. Thank you. Jeez.
I'm just going to exercise those upper swing arms a
Tough
bit,
one.
Sean.
Yeah, no, go for it.
We also have Dr.
Pilot,
John
go ahead.
Smith from
Yeah,
the
if you wanna get a move
Oceanography
in,
Department
I
at
think we
School of
could do 50
Ocean
meters.
Earth Science Technology at University
No
of
problem.
Hawaii. Megan, who just gave that
Anything from 40
awful
to 60, I
joke.
think is fine.
Of
But
course,
we got to
Nicola
20.2.
Rodriguez, who's next to me from the University of Hilo, Scott France, University of Louisiana at Lafayette, and
Video,
Tim
you wanna
Shank
come in on
from
this?
the Woodsole
Covered ground
Oceanographic
over this.
Institution.
Yeah, it's
Sandy
pretty
We'll get more as the
sandy
day
and
goes
great.
on, typically. So
Flat here.
thank you all for joining. And we got a drunken shrimp.
All right, I'll just call in a big move.
And
Yeah,
what was
Washley, it's hey, we're on bottom, and we're ready to start.
Great.
Excellent.
well, we're gonna get a view of the shrimp to start off with, mostly because it's the only game in town at the moment.
Yeah, we're putting a ship move in now. We're going to try to move past this sandy bottom pretty quick.
It's fine, you know. It will be on fish watch, I think.
Copy that.
Fish watch.
Fish watch.
You gave us the name of this
All
shrimp
right, I'll still get us
yesterday, I
going.
think, right?
Yeah,
Yeah,
it's a
definitely.
Canthophyra.
Bridge now.
Canthophyra.
Just beware, Sean. I'm looking at 050.
That was
for the moment.
confirmed. I'm not positive by who
Once I'm done with this, I'll
yesterday.
turn my
Hey,
head.
we'd like to get a ship move if you're ready.
Copy that.
Send it.
50 meters, bearing 050, speed 0.2.
Ship move range 50 meters at a bearing of 050 degrees.
That
Decimal
is
2 knots.
snappy.
Good copy.
So I'd have to look back in the event log, but they suggested even a Canthophyra
Right.
eczema, which is a species that's known to occur here in the Central
Okay,
Pacific.
based on the multi-beam,
Thank
this
you.
move should get us to the
Yeah,
base of the
Bridge.
slope.
So,
I think
Thank you,
we're
Bridge.
good.
Money come wide.
So,
Let us proceed.
All right, Sean.
All right, happy flights. Look good.
Bring my
good.
head over to 060.
Head full.
I think we're on 50.
Oh, you
actually.
put it in 050?
I asked for
you find
50.
sea
Gotcha.
stars on
Okay, we'll do
sediment,
that
don't
then.
you?
Oh, yes, absolutely.
Oh, is that a fish?
Seeing no
Is that a
terrain
fish
in any
or
direction.
Something is in the water column
Upper
there.
right-hand
50.
corner, some
Yeah.
kind of white
That's
sinusoidal
that's to the
body.
waypoint.
All right.
Yeah,
Got a
watch it. This is
feeling.
as close as I can get right now, but we'll see.
It is a fish. All right, Bruce, you're on. How about that for delivering right out of the gate?
Okay, thank you for taking that request. You guys are really good. So we're a little distant. This is a halosaur. We can't say what genus yet. We need to look at the top of the head to see if it's scaled or not. But my suspicion is that it's an Aldrovandia species. And this one looks like it's pale
the track is only about
instead of
500
dark
meters.
brown or
Yep.
black.
So we've got
Which,
time.
and it has
Take
a
our
long
time,
snout.
get a lot of
and
good looks at
trying
everything
to
we
remember,
see.
Ken Sulak had said that these might be
So we're done with this,
Aldrovandia
Sean. Maybe we,
rostris, I believe.
Come back and we can kind of look
The
to the
pale
sides.
white ones. It's a
Zigzagging.
good distance away, so I
Copy
know that the ROV pilots are going to have trouble acquiring a good image. If it's this Aldrovandia rostris, that would be a new species record for the Johnston Atoll area. The taxonomy is a little uncertain because there have only been collections of these around the Hawaiian Islands and not down in Johnston Atoll or other Central Pacific islands. And it's
Copilot,
difficult
when
to
you have a
identify
minute,
the
can you
species
go
without
ahead
a specimen
and
in
set
hand.
DC bus
The
return
reason I
ground
was
fault
hopeful
monitor standby
for
to
seeing
disable
fish
it. Why?
on
It's
this
supposed to
at the
be
beginning of
there.
this dive and the soft
Indeed, I will
sediment
turn
is
off
that this fish and a lot of
the
others feed on
ground fault monitor
crustaceans
standby.
and worms,
Just to turn
polychaete
on
worms
the device.
that live
On the
in the
DC bus
sediment
return.
and
Yes.
emerge
Thank you, Copilot.
at various times
And we will
to
record
forage
values.
for food. And so the food supply for fish like this
Once it
would be more
updates.
abundant in the lower gradient soft sediment habitats than it would be on the high gradient,
think it's about time to
very
give up
rocky
on him,
habitats
pilot.
that we've
Yeah,
been
when he can
exploring
wipe
in the
it, yeah.
last few days. So a halosaur. Thank you very much to the pilots for trying
65,000
that. I hope we'll
kms.
see more.
That's
That's a little
If
high.
you guys are
It's the
taking
max of
requests,
a
can I
yeah.
ask for a bamboo coral
It only
actively
measures to
feeding,
50.
please?
Sure, we'll deliver that right up.
A bamboo coral actively feeding. Okay, we'll look for it.
Well, first, here's sand. Is this a... What's this... Is it a white... Is it a cucumber, or is it... There's a white body ahead of us.
1300, that's good value.
Yeah, copy that. Watch
Is it a thing, or is it just...
Are you writing it down, Dave, or do you want me to over here?
Why don't you? That's more appropriate there.
All right.
I'm writing it down as it
Okay.
affects
It's-
CTD data over here.
Oh.
But
You
not,
get your
not engineering.
imagination diving away with
I
you.
think it might just be. Wait, video quick sim.
Nobody knows that song anymore. I guess that
Oh.
ages. Oh no, you're right. Oh, I can't believe it. You are right.
I knew my Okeanos sense was tingling.
Wow, look at this guy.
Wow, look how well disguised it is too.
Yeah.
So this is a sea cucumber. You'll hear me repeat this little mantra over and over again, but these are kind of the earthworms of the sea, but they're echinoderms related to sea stars and sea urchins. They essentially devour sediment and eat all of the organic goo that's present in it. They devour that, they ingest that goo, and then the sediment just is sort of deposited out as
request
castings.
was for lasers off.
Kind
Lasers
of
off.
like the way that earthworms do. This sounds kind of gross to most people, but it's actually quite an important ecological function because they can process the microbial flora in the sand or sediment, and they can also, and this also frees up the sediment for other animals that burrow through it. So sea cucumbers actually have quite an important role in the carbon cycling and the sort of geochemical cycles.
Caught that one. Is that Chris Ma?
In habitats like this.
I wonder why it's
And this one seems to be well disguised. I'm not sure if that's the skin or genuinely the color of the skin, or if it's simply covered by sediment as kind of a defense. Sometimes sea cucumbers are able to use the tube feet that they have on the surface to adhere sediment and other particles to their body.
So there's only one type of
Exactly the question I was just raised with you, Chris.
Oh, what? Sorry?
I'd say that was exactly the question I was just going to raise with you, whether this was one of the sea cucumbers that might be using its podia to disguise itself. It does kind of look like chicken breast kind of rolled around.
All right, ship is well underway, and you should be swinging.
I know exactly what you mean. Yeah, it's hard to say without sort of
pretty far out there and kind of pulling my tether here when you're turning around.
Animal. I'm sorry, Chris.
The only one that I'm familiar with that decorator holothurians, if you will, is Mesotheria, but I don't know if that's it or not. And that's in the Synallactidae, so it's sort of a weird Synallactid.
It's hard to say. I mean, there's no characteristics to see here.
I know you can't see the feet or anything.
Yeah. But we're done.
Right,
so whenever video is ready,
how long ago did you put in the move, Nav?
Yeah, video come on when you're ready.
A move was 6 minutes ago. The ship has moved.
I like that.
Okay.
Chicken
30
breast
meters already. We have 20 left. And you should be
well, some of them actually have kind of a cat-like behavior where they kind of roll back and forth. It's really strange, but I've seen them do it on videos. It does literally seem like they're battering themselves up.
Yeah, I guess that would beg the question in this particular habitat, what would it be camouflaging itself from? So perhaps that's not an intentional coating of sediments, but just because of the amount of mucus that's produced, perhaps, if that is actually sediment on the surface and not the color of the tissue.
Yeah, I mean, for all I know, it protects itself against something like those halosaurs. I don't know if there are very many visual predators at this depth, but a curious defense mechanism, to be sure. If that's indeed what it is.
Pilot, could we get the lasers on, please?
Lasers back on.
Copy that. Lasers on.
Great, thank you.
So I'll try to be good about turning those off when we have a nice beauty shot.
So we always tell everybody, or remind everybody, that the two red dots you're seeing in the center of the screen are 10 centimeters apart. And that's how we measure things down here and get an idea of their sizes.
What is that white? Is that a rock?
What?
Oh,
about
sorry. There's a white
right.
circle that we passed.
Yeah, copy.
And
some
just
rocks up
wanted to
there
see
ahead
real
on
quickly
Sirius.
because it's not the color of the rocks and it's round.
But I am seeing rocks in the background there.
Yeah, it's right there. Yeah, so just down. Do you see it right?
Yeah, I see it.
Yeah.
Sharp eyes, Chris. Oh, look at that.
Coming back.
Now all we have to figure out is what phylum it belongs in.
Huh.
Is this one of these holothuroidians that's buried or something?
1, 2, 3, 4, 5, 6-part symmetry.
There are certainly sea cucumbers that do bury themselves in sediment, so that is a possibility.
Nah,
I don't
Bridge,
know. It has
complete.
6
Thank you,
arms,
Bridge.
and it looks like its base is actually above the sediment. I mean, it could be an egg, a discarded egg case, or weird.
Yeah.
Yeah, I don't think it's alive anymore.
Looks like it has a prominent opening at one side.
Yeah. Huh. Well, I don't think it's alive anymore in any case. So
Might
we
have
may
enough tether
discover
to get
what
up to those rocks
hatched
pretty
from
soon.
this.
Just a couple minutes.
If anything.
Yeah,
All right. Thank you. We can move on.
All right, video when you're ready, come
Appreciate
live.
it.
Undetermined. There we go. We have an undetermined category.
Gelatinous.
Whatever. That was a weird one. Sharp eyes, Chris. Almost looks like a weird anemone the way it kind of opened up.
Yeah.
The only thing that makes me think it's alive, it was closed when we first saw it, and then it looks like it opened up its arms or something.
Yeah. There's a lot of weird stuff that I'm sure we'll discover, and we'll just never see it again.
So overall, this is not a very tall,
So,
prominent
pilot,
feature,
before
is
we
it?
get too far up the wall here, if there's any decent rocks, this might be a chance to pick something up.
Yeah, copy that. If any stand out, just let me know.
Okay, thank you.
All right.
So, about 100-meter feature, 150 meters.
Okay. So I'm looking at 060, and I'm getting some pretty nice returns on sonar now.
Copy.
Okay.
I think we just have a couple minutes left to swing, but sounds like I'll look for a rock here potentially. So
There's something small above the bottom just going to the right of the laser.
Well, I saw something float by just
did
a
you?
moment ago. Yeah.
Is it really small?
Could be a little shrimp or mysis.
Yeah. That's okay. We can move on, I think.
Copy that.
Yeah. Especially since we'll be hitting some rocks soon.
Like right now. Look at that juicy monster over to the right. Maybe a little big.
Yeah, we're going to have to pass on that one.
Is there
Right.
something? That's a shadow.
we're getting better at that.
It's possibly one in the background upper left, pilot, above the lasers a ways. I wonder, we're not there yet. It may get to be enormous when we get there, but it had this girth and it looks loose.
There's a sponge on the big boulder to the right.
I didn't even see that. Where?
Oh, it's off screen now.
It's off screen.
Yeah.
Where are the lasers? That pilot, is this one coming up the center? Is that too big, or would that actually work?
Coming up, so
Yeah,
further
it's
past lasers.
It's about 10 o'clock now. I mean, it's big, but is it too big is the question.
Let's see. So that looks pretty big.
Okay, how about the one to the left going off screen almost right now? Again, I'm just looking for something with a little bit of 3D-ism.
Yeah, copy that.
How much left in the move, Nav?
We're done
Where's the
moving.
fish?
Well, we have
How
it
much
left.
swing do we
Enough.
have left?
Maybe we could look for the fish. Oh, there he is. He's above that
probably
rock.
5 meters if that.
Okay.
Video
Yeah.
zoom on fish.
Oh, it's a different one, Bruce. Got a different fish.
That 50 move may have been a little
Yeah,
aggressive.
so this is a fish in a different family than we saw. The other fish is
I'm only gonna
distant
swing a
relative
few more
to the
meters,
true
it's fine.
eels. This fish is a grenadier. They're also called rat tails in the family Macruridae. Very difficult to identify to genus or species. This one has kind of a bulging stomach that's a little bit different than others that we've seen. May indicate that there's good food here for these little fish. It's in the family Macruridae. So the grenadier rat tail, and unfortunately from this distance we can't tell what it is. I need to make a correction on what I said about that halosaur, too. I threw out the name Longirostris. Actually, the name should have been rostrata for potential species identification. Rostrata refers to the snout. That one had a relatively long snout. This one could be a small Coryphonoides. Coryphonoides are active predators and scavengers. This one's kind of drifting slowly. You see it's got a long whisker on its chin, and it's got fairly long rays on the front of the fins on the belly, the pelvic fins.
Bruce,
the tail's a little long, but there are several species. Chris Kelly, what do you think about the potential genus?
Yeah, are you seeing how high the dorsal fin is on this thing? I'm just curious about that. It's higher. I mean, Coryphonoides can have pretty long dorsals, but is this another feature? I certainly see what you're talking about.
There are a number of species in Coryphonoides. This would be not one of the large ones that we see, it's not Coryphonoides longiceros or Coryphonoides armatus or equine. It could be a different genus, though. There are a number of genera. It's not the small one that we see frequently at these depths called Kumba. It's something else. So we'll have to try to send images off to some experts, some people working in California, in Virginia, and also Japan and New Zealand, to see if we can get a suggestion about the identification. I don't think we've seen this species before on our dives, at least the ones that I participated in. So it's very interesting. A grenadier in the family Macruridae, the distant relative of the codfishes. And thank you for the videography. That's great.
Thank you very much, Bruce. Appreciate it.
Thank you. We can move on.
Co-pilot
There's actually
down
a coral to the left. I don't know
there.
if we're in a position.
Yeah, I saw that earlier.
All right, video, come on.
It's been hanging out there, though.
Yeah, that's more the value I expected. That
Coral to the
1300
left.
was surprising.
Yeah, it was where the...
Is it back towards the rock? I try not to drift too far away.
There's a lot of other rocks up ahead of us if they're not.
It was right around where we saw the fish, but I think it was in front of that big boulder.
I see one on the right side of the rock.
We're all settled out.
Right offside the screen.
That's it.
I'm surprised I'm not seeing a bunch of sea pens in these sediments.
I thought there is one right here.
Yeah.
Scott, stand by. You might have called something already before we got there. It's a little stock thing in front of the bigger boulder in the upper left.
Yeah, I think that's what I was seeing earlier.
No, because I'm delayed, I'm referring to something that was on a rock
12
seconds ago, 14 seconds in the other direction. So that's okay.
I'm not sure it's a pen. It does sort of look arborescent, doesn't it?
I'm pretty sure it's an octocoral of some kind.
Yeah. No, I mean not a sea pen, maybe.
Oh, it is a sea pen.
Oh, it is a sea pen. That's just a shadow.
Ah, okay. Oh, interesting. Ah. Sneaky devil.
I'm not going to be able to do anything more than sea pen.
Yeah, there's not really enough to see.
Yeah, sea pen, Chris, I agree. Maybe Cophobelemnon, Calibelemnon, something like that. These are, they can be pretty tricky to identify from the images if we don't already have a good sense of what we're seeing. But just to provide a bit of information, since we're looking at it, they are indeed octocorals, as Chris Ma has said. They're specialized for living in soft sediments. That's why I said a few minutes ago I was expecting to see some. The base of the colony has an inflated portion that serves to anchor it into the sediments. And some of these sea pens actually create kind of a burrow. And so if you were to reach out to try to collect it, some species, not necessarily this one, but some could completely withdraw. It does have a hard axial skeleton in there. It's completely unbranched. It's just basically one rod. And around that one rod, the full length of what you see in the colony, that is a single polyp, and then we refer to the secondary polyps arising from it. And I heard you yesterday or the day before saying one of the ways that we can distinguish among the different types of sea pens is that those polyps arising directly from the main primary polyps, or if they come out on little leaves or fleshy arms of tissue and rise that way. These ones, the polyps are coming out directly. Thanks for the close-up.
Yeah, thank you, Scott, for the information about it. I think we're good, pilot. Whenever video's good.
And then do you still want to look at that rock?
Yeah, we just
turn
the
my
one
head towards you,
behind the
Sean.
C-pen,
Yeah,
it's got some angles to it. Is there any way you could grab it without disturbing the
seriously,
pen?
on a walkabout.
Because it's a nice angle. Well, it might be a little
Jesus.
small,
Say it
though.
again. Sorry, Washer. Which rock are you referencing?
It might be a little small. I saw an angular one behind the pen, but I think it's on the small side.
Copy that.
And if
The
I
one
can
you-
follow up, that boulder behind the C-pen has a white circle on it, on
Copy
the
that.
darker half behind it.
Do you want to see the rock before? I think I'm right on top of it. I can back up.
Sorry, yeah,
there,
no, that's
or
fine.
How
whatever
about,
order you want. It's fine with me.
well, excuse me, pilot, whichever one you want to look at, but there's some rocks to the left of that boulder that might be in that sweet size zone of around 20 centimeters or so across.
Turn in my head,
Copy
Sean.
that. Copy that, pilot.
Maybe the one all the way to the left there, just,
Right to left.
well, yeah. It's the bigger one, of course. I'm asking for that one now down straight from the lasers.
Oh, it's on screen.
And to the right of the lasers. Keep going a little bit further.
Yeah, that one right there.
That one right there.
That one we could probably do if it's loose.
Yeah, John Smith, are you on the telecom right now? Or Jonathan Tree? Well, let's go ahead and get a look at this. I think this will work.
Yeah, copy that.
Yeah, that's a real good size rock. We kind of try to get girthier rocks that could have basalt inside of them, just because all these rocks are crusted with manganese and other minerals, and sometimes that's all there is, just crust on it without any basalt, and it renders the rock useless for geologic aging and geochemical analyses. But if it's a nice girthy rock, there's a chance the core of it has basalt in it. Yeah, that's a nice one, pilot. Why don't we go ahead and grab that one as the first one if you can.
Yeah, copy that. Just get settled. All right, Washies, we're just going to do a quick chair swap.
Great.
20 seconds.
Yeah, no problem. Thank you. We're going to do a quick swap. They do this to give all of the various ROV team opportunities to work the manipulator. And
Looks stable there. Co-pilot, do we have hydraulics up?
Are you set up?
I think so.
Okay.
yeah. Hello, John. Is that you?
Yes. Yeah.
Are you looking at this rock? Will this suffice?
Yeah, the one with the lasers on it.
Yeah, correct.
All right, can you grab Mini Zeus?
Yeah, it looks
Sure.
good.
You want me to put up on the big
Okay,
screen?
that's our target.
Sure.
Okay.
It's a reasonable size to put in the box. Looks like it's got some height to it too.
And I don't need help to deploy.
There's also one in the upper right corner of the frame. That one would look good too. It's kind of angular.
It might be a little smaller, John. We were kind of fooled by a bunch of smaller ones that looked.
Do the little nudge test.
Okay.
By the way, while it's free.
Oh, that'll
Good.
move.
While he's getting ready to grab this thing, John, I just wanted to report that we got a look at the smaller second rock that we collected yesterday.
Okay, I'm
The
gonna,
first rock
this will
was
fit
very
port
heavy,
side,
so it's
I'm
got
gonna
something
open up.
inside of it. But the second one
All right.
The
was
rock
amazing
rock.
Sure.
in my mind. It clearly was a pillow fragment, and it clearly showed
Follow me
vesicular basalt
bring the
without
camera down
that
to
much
the
manganese
wings so I can see.
crusting
wing
on it.
Okay.
Yeah. Makes it
Sure.
much easier.
Oh,
I
that's
agree. I'll get out of the way.
pretty interesting.
Yeah, you could very clearly see all the little holes in what we. Maybe you can tell people what vesicular basalt is. What am I saying there anyway?
That's plenty.
I've got a bunch of
just a lot of holes in it. And basically, there's gas, bubbles, frozen in the rock. So it means the magma had more gas in it when it came up, and that's interesting in the underwater realm because you have the pressure of the water usually keeps those from forming, is more restricted in formation than it's erupting on land. So then that would indicate that it's either a really gassy magma or maybe a shallower eruption of it.
Yeah, what caught my eye, John, is the fact that
You kind of got it in the coral cutters there.
this was on a seamount that showed no indication of ever been at the surface. There was no flat top or anything as part of it. Okay, we've got this rock. We're going to do a little spin here first. And see if we can see any commensals on it.
Video, can you come in on this?
I just want to let you know we have taken from roughly squall off of our bow,
right?
so we might have to be adjusting a heading sometime.
That's correct.
Copy that. Yeah, just keep me posted.
Okay, we'll do.
Thanks.
No, clear.
So yeah, it could have been a pulse of different.
Pretty fine sediment coming off of it.
Magma.
There's something, pilot, excuse me, to the lower right. Something white on the bottom of the rock.
Lower right. Okay, I'm looking at something, or potentially something top center. Is that?
Oh, okay. Sorry.
That might not be real. I don't think so.
Since you mentioned that white thing on the rock, did you happen to get the hydrozoan that was on the rock yesterday?
Sorry, Scott, because that was in the rock box. He was gone.
Yeah, that's what I thought might happen. It sounds like you had a late night, but I'll ask anyway. Were you able to look at sclerites of that white thing? We wondered if it was a Paragorgia.
Video, do you have any more Zoom?
No, I'm afraid not. I didn't even look at the sponge, but I'm going to tell you about the sponge after the rock is tucked away.
Interesting.
Okay. It looks like this has a lot of those little tree-like things we've been calling forams, arborescent forams, so that'll be really fun to see. Happy picking.
Yep.
We'll save them for you, Scott.
Thank you, Video. We're good.
All right.
Yeah, I picked lots of those off on an earlier cruise this year. I was quite diligent.
Hey, Chris, you know it's a good idea to leave the rock on the ship for Scott.
Wrote about prior to our cruise that are only less than a dime in size, and they were quite eager to see some of those.
Beautiful. Thanks very much, pilot. Great grab.
So you can either close it manually or I can just
okay,
bring the
so
wing in and
that's rock one, specimen one. We'll get another rock and a couple, a few biologics too, if we can. But I want to tell you about the sponge. So when the ROV got on deck yesterday, and this is a yellow presumed polyopogon sponge. It had the fibrous attachment to the dead coral, as you recall. And it was one of those sponges that was landing on all of the dead corals very curiously and not on the rocks. So we grabbed the sponge, we got the whole thing up, opened up the ROV box, and the water was blood red, almost amber, and the sponge was sort of a little bit whiter, paler color. We put it in the bucket of seawater, cold seawater, got it in the lab, and it turned the entire bucket of seawater blood red. We were able to remove the sponge. It was now quite white, and we put the sponge in 95% ethanol. It was small enough where we decided to sacrifice a little bit of ethanol and put the whole thing in it. And now the sponge is absolutely black, and the ethanol had turned deep dark purple, and we saved a small sample, of the
So you're
ethanol
need to
with the
come
purple
way to
color,
starboard.
but I can also
Alright.
tell you
I turn
from
my head
two of
about
us
90
that
degrees there.
no matter if you
Alright,
got
please
gloves
use
on
my
or
gear.
not, you get a little bit of that water on you. And we both felt some stinging from the water, and it was either, we think it's hydroids, but we're not positive. I think he had some hydroids on there, but it's very curious
Oh, yeah, Nav, I was wondering,
what
you
that
cleaned
collection,
box out
but I've
last
never
night,
seen a
right?
sponge do that
With
before.
the
And
red water in
that
it?
sure is a good indication
no,
that
it's
we
not
got
me.
something
Oh,
brand new.
We hosted out pretty well.
Okay. I was just wondering.
Chris,
if
that's
we ever got
really
any
interesting.
reaction.
And I'll tell you about some corals, but just before I do, the large boulder you're leaving on the left has a little something standing on the top of it.
Say, pilot, there's a white
There are plexor.
body.
It's okay.
on
It was a little stock of something.
Okay, we can move on, pilot. Thank you.
There are plexor corals that behave
keep
exactly
them
the way
starboard.
you just described, in that they are yellow in appearance when you see them in situ, and upon putting them into ethanol, the pigment that's in the tissue leaches out and turns
Yeah, you
the
need to turn to
ethanol
starboard.
fluid kind of
turn
a
as
black,
well.
deep
Okay.
purple,
I
and
need
the
to
colony
see it here.
becomes either very dark or black in color. So I wonder if the pigments will be the same kind of pigment.
A little bit more of a
So
feature
Scott, I don't know if you remember the yellow plexaurid that we collected a couple days ago, maybe. And that plexaurid is exactly what you are talking about. It turned completely black in ethanol, and that's what it looks like now, completely black when it was bright yellow when it came up.
Yeah, very interesting. Doesn't surprise me at all, and is certainly something that we've seen. And it does, as you just said, maybe that indicates that sponge is a new species, and what you're doing is you're using the presence of a certain kind of pigment to denote that that sponge is quite different in terms of its physiology from other sponges, and that's one of the ways that we can identify the corals as well. Not as well as the sponges, and in the sponge field, there's a whole field called chemotaxonomy, where they use the presence of pigments and other
Yeah, I
chemicals
see that. You're at
to help
7.5
sort
now,
out
almost
their
8.0.
relationship.
Yeah.
Yeah, thank
Yeah, I
you,
think
Scott.
Video come in?
One more pass here. 075.
7.5 you're happy with?
Polychaete
Yeah.
worm, I
Copy
think.
that.
Is that right, Chris?
Do we want to wait a little bit, or?
Yeah, we can wait.
Copy.
We're not in a rush today.
Swimmy.
You think this is a species of swimmer?
Oh, no, I was just...
But it's swimming, right? I mean, it looks like it. I wasn't joking about
It's hard
this.
to tell, really. But it could be, certainly. Swimma is a genus of polychaete worm, a bristle worm with that can release little sort of glow-in-the-dark bombs to confuse its prey. But it's hard to tell from this far away.
It's changing
Oh, wow.
direction.
That's nice. It floats. There was also a branching structure on the rock that was just below us.
All right, we can go to that.
I think it was further. Yeah, we might have passed it. That's okay. We can move on.
So pilot,
Yes.
because we have a short track line today, we can definitely kind of move back and forth, side to side,
All right.
and zigzag up the slope leisurely.
Those of you just joining us, you're watching a live broadcast from the NOAA ship Okeanos Explorer.
Video switching in.
We are in the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument, and we're in a seamount that just got mapped actually a few hours ago, and we had to very quickly pick a dive site on the seamount, but it had never been mapped before, and it most definitely has never been dove on
Turn in my
before,
head to follow you a bit.
and I
Okay.
don't believe it's ever been dredged for rocks before. So this is real, absolute, true exploration. We're seeing things here for the very first time, and you're seeing it with us. Our cruise, this particular cruise, we named Laulima Oka Moana, and it's really denoting
at this point.
partnership in the sea
I
because
just want to
we have an immense partnership
A little bit over here.
to actually carry out these
Some
cruises
texture on
and
the
these
rock, all
dives between a whole different bunch of
So
organizations, a lot of NOAA line offices, as well as the Global Foundation for Ocean Exploration, University of Hawaii,
Okay, you're getting out there pretty far
the
forward.
Smithsonian Institution, all the
Why
participating
don't we push to starboard
institutions
and
that our participants who are
another
actively
pass here.
participating
something
are
under
from.
this rock.
Okay.
Video, can you come in? It's going to be in the
So
shadows.
that's what we're doing to provide you with
Move that
absolutely unique footage and unique exploration of the deep sea floor.
Now,
And we are currently at a depth of 2,000, almost 3,300 meters, 2,295 meters down below the sea surface, so well over a mile below the sea surface.
Looks like a worm tube in view, some kind of sediment tube.
looks like it's empty. All right, come on out with you.
A very textured rock surface is manganese crusting that coats a lot of the rocks at these depths.
Now, if I'm not mistaken, that's called botryoidal
Botryoidal.
texture.
Yeah, I like that word.
Yeah,
Pretty
it's got a
cool.
nice roundness to it.
Yeah, and you
Come in
can
underwater,
clearly
I
see
think
this
it's
is
not one of these
volcanic, or at
worms.
least John Smith and other geologists can.
Here's another swimming polychaete coming
Where
up
is it?
to us.
Ah, okay.
I'll just let it go.
Yeah, I'm going to let it go.
Thank you very much. So we are coming up on the lee side of this kind of slightly ridge-shaped summit of this seamount, which does not have a name yet. And typically on the,
Something on the
we
rock here.
think it's the lee side. Typically we see more sedimentation, which is quite possible what we're seeing. And then when we get up onto the crest, we see it's a little more current swept and a little bit clearer consolidated rock. And this is where we think we're going to find more corals.
Is that nematocarcinus?
Yep, nematocarcinus, our old buddy.
That's Maxim.
We're good for Science Pilot. This is kind of a regular denizen of the deep.
Okay, come on out.
Can actually spot these things from like that distance, because they're the. After a while, you get a gestalt for them, and they're everywhere.
I think one thing that the deep sea
Is there
hasn't
something
failed to
in the
deliver
sand
is a nice
now
array of
directly below the
terrains and
lasers?
handsome
Let's find out.
landscapes for us to look at while we're searching for animals.
No, just sand.
Just sand.
I'm glad you said that, Chris. I'm often not sure when I
This is something.
tell people about the deep sea. One of the things that comes up a lot is that it's a fairly homogeneous habitat. And what that's really referring to is the fact that the temperature really doesn't change very much regardless of where in the ocean you are. It's all
Come on in.
typically below about 4 degrees Celsius. There's not much light, it's high pressure. But at this scale, at the habitat scale and the scale of the organism, in fact, there's a lot of heterogeneity.
Yes. That might have actually been a sea cucumber, but I'm not sure.
Oh, was
so-
it? Okay.
Oh no, that's fine. It's
I thought it was initially, but we zoomed in and.
yeah,
and,
it might have
changed
it
my
might
mind.
have been. It's hard to say. There is lots of little patchiness in terms of sort of lighter colored sediment.
All right,
I
pilots,
suspect that
are you
those
still happy, or
are
do you wanna
where different kinds of burrowing
more?
animals
Probably.
have been
think
hard at work
maybe we should do another-
feeding on
just
the
go out to the
organic
starboard
goo
side.
that's
Copy.
in the
We're-
sediment here.
Our average pace needs to be 0.05 knots today.
There's a
so
sea cucumber, isn't it? The one right above the lasers right now?
Let's
It's-
take a zoom in on that round.
Yeah, there's
It might
something
be
there.
that Antipitherian that creates hoops around itself. It looks like a just a little lower
Yep.
when you get a chance. It looked like it was, or was that an illusion?
It's an illusion. Oh, okay, never
I thought I
mind.
saw something up in the water casting a shadow.
Yeah,
Yeah.
I thought I did
I mean,
too.
This
Sure
was
look like
moving.
eating trees.
Oh
Yeah,
no, see there it
is.
is.
it moves.
Is this something that's just rolling back and forth, or is it actually
That's
part of an
Maxim.
animal?
It looks like it's a stock, and it's coming out of this hole, and it looks like
set
it's
down
rolling
if you
back
wanna
and
come out.
forth. And look, it's another one of those funny egg-shaped things. Those open. Huh. That's an interesting.
We're gonna set down and try to get a better view of that.
Yeah.
Come back in.
Yeah, see, it's coming out of a. Whoopsie.
So, pilot over there, that black hole that's upper left. It's got a little fleshy stuff around it. Could
Okay.
you zoom in on that, please? This is one of these curious beasts that we were trying to figure out whether it's alive or dead.
Do you have any more video? We're in pretty close. This is very small.
Is it? What
Well, it looks
are we?
organic, at least.
Yeah, I think it's alive.
Yeah, it looks like it has some kind of reticulate root or holdfast. And
That's
then
Maxim.
the top is something.
So, Scott or anybody out there, has anyone seen something like this? Let me check the chat room here.
Yeah, it's curious, isn't it? I'm wondering if it's some kind of mollusk, and that's a very elaborate mantle edge. Sure has a big opening there.
Yeah, it sure does. Okay.
Well, the question, too, is whether it's the same thing that we saw with that kind of weird six-pronged opening.
I think it might have been, because there was sort of a hole on that thing, too, if you remember.
It was an inside hole
That's,
after the prongs had opened.
that's true.
Yeah,
Interesting.
whenever you're happy, let's get it with lasers.
Yeah, we can
Yeah,
move on.
we're good.
All right, video
Thank you,
coming
Colin.
up.
Sirius
Nice
is
to have a
not
bivalve.
tracking well.
Well, that's true.
No,
That could
not
be
at all.
something that's buried. That's,
Yeah, that's true. Could
So
be.
it's pretty small.
Can we do a noise
Yeah.
test right now? Or
I just did.
There was one.
Okay, so maybe we can just go ahead and move on.
I can do another
Pilot.
one, but
Okay.
I think we're
I
good
did
with
5
that.
yesterday, and Fernando did
Come
3.
on out.
okay.
So 8 total and 0 correlation whatsoever.
Yeah, I haven't
Really?
noticed any
Okay.
correlation.
All right. Well, why don't you kind of
Those linear structures seem to
back.
be around. I bet we'll see
come to
another one.
starboard?
Those things that look like bottle brushes.
Okay.
Probably another shrimp down there. There's something on the rock. Becoming partial.
Ah.
A Xeno, huh?
Yeah.
A little bit more.
There's another nice one on the. Oh no, this is one. Let's take a look, a closer look.
Could be a dead stump or something,
yeah.
too.
I think this is another one
better
of those
to the
6.
right.
Is it a 6? I'm not sure what it is, but yeah, it could be a foram, some kind of xenophyophorian test. There's also to the. Oh yeah, okay. So there's another one that's sort of similar to it. So these look like the sediment structures left over or created by single-celled organisms called xenophyophorians, or possibly, what's the other one, coccomacians. But basically they're giant amoebas. There's another structure over to the right that is more like a fan. I think if we pull out, we saw one coming in.
Okay.
There's a sediment structure
Come on out,
that
video. Try to find it.
All right, Sean.
that basically just looks like a
We could
little
probably
tree.
do like 20
And those
meters
are
towards 070.
the same types of things.
20
left
meters,
over by these
070
animals,
I.
or these single-celled creatures.
Yeah, there's something right on the left side of this boulder, Pilot. Could be a crinoid or
It's,
something.
moving toward it now.
Oh wow!
Oh,
look at
wait
that
a
big.
second.
There's a
No, wait. Oh boy. Is this one of these Cereanthids?
Come on in.
This
see.
huge? Okay, Pilot, this is a collectible. And this is a really tricky collectible. We found out the other day it swims. believe it or not.
Really?
Yes.
Looks pretty well attached.
It won't be.
Okay.
So I think, Dan, I think you're over there. Is that right? Dan Copilot, you remember this?
I don't think I saw this one, no.
Oh, this is on, I think, one of the first dives. We tried to grab one of these very curious creatures, and it swam away, and we didn't get him.
I think maybe I saw a clip of it, though.
I've actually been looking for one of these since that day.
But we're
because
moving the ship, Dave, so why don't you come back a little
it doesn't
bit?
make any
Okay.
sense. We think
we'll find
Tina
it.
Molitsova thinks it's a Seriantid based on oral tentacles, which you can't really see from
Should we
here.
get,
But it
shots
makes no
of this?
sense because it has no tube and it's on a rock.
I can, turn off the laser
So
switch.
we don't even,
wanna
we're not
practice
even
your
sure
pure
what
wet skills,
phylum,
the ship will be up
excuse me,
there
what
shortly.
order or class,
Sure.
I guess, of Nidarian. Yeah, let's get some good close-ups of this. Expect it to swim away
Chris,
when you
if
try to grab it.
This is a Seriantid on the other side of the rock, that face that you can't see. There should be a tube.
That's Maxim.
And when we did a cruise back in 2004 in the Aleutians, or 2005, one of the two, we did see a Seriantid tube anemone on a rock. Its tube was glued to the rock, and we were able to collect it because the rock was loose, so we could pick up the entire rock with it. But otherwise, I think it would be really hard to try to grab this thing.
Well, they just got to prepare for it to swim. We almost got it the first time, and it does not have a tube stock. Scott, I can absolutely assure you it has no tube. But it's got that inner oral tentacles all around it, which you can't really see at the moment.
Okay, let me go. Let me know if I'm getting into trouble with that swing arm.
Yeah, absolutely.
Thank you.
Looks fine right now.
Yeah, Tina is saying is okay, they can live without tubes, so thank you very much. Tina Molitsova is now wide awake, even though it's late in Moscow. Probably her eyes popped wide open when she saw this
What
thing.
time are you calling the move, Sean?
So thank you. Tina's from the Shirshov Institute in Moscow.
Only 2 minutes ago.
Okay.
Yeah, so you're not going to
Well,
feel
it does
anything
have that
for
inner
maybe
row
another
of,
2 minutes.
Yeah, so we're gonna try
Yeah,
this.
if we can look it down the center of it, that'd be great.
Okay, I'm still
Yeah.
We're just gonna
rotating around.
All right,
turn
now,
my head a
pilot,
bit
you
toward you.
careful with your light pole now.
Watch your tether, because you're gonna start tugging me.
Yep, I did.
We're not gonna be moving for another
Come
couple
out,
minutes. Yeah, you need to come to
That's the end
starboard.
of the tether.
Come to starboard. Yep. We'll be there soon enough. Just give it a couple minutes.
Yeah, pilot, if you go back to that position once you have tether,
I'm trying to figure out
might
how
wanna
you're
pick
going
up
to do
your
this, pilot,
port
because he's
lower,
on the left side of the
'cause it
boulder
got-
and the arm's on
I
the
mean, it
right
was still
side
safe,
of the
but-
ROV.
Yeah,
Yeah.
I think we'll figure it out when we get a little more tether. We can get closer.
Sure.
But I think we can do it by setting up sideways and looking directly at it.
Okay, thank you. This is one of the strangest mysteries that we've seen in the 3 years of the Capstone expedition. That's why this is worth a little bit of time here. I mean, when you can't even go to class or order or something like that, then you really don't know what you're looking at. Yes. Thank you.
So we'll go ahead and bring in that swing arm now.
Okay.
Yeah, it's a good preemptive thought.
There we go.
Also, could I make just a suggestion, even though this is your specialty? If you had the box open and ready to go, you can almost, it was Sean, wasn't it? Are you over there in nav position,
Yes.
Sean? Was it, you were the one who was trying to get this, right?
Video calling again.
When was it?
We've got a little
This
more
was
tether
like
now,
the
Dave.
first dive, I think. Remember the big, huge anemone-like thing that swam away? And then
I don't think so.
I'm trying to think who was on the stick at that time. It was Chris Ritter then. Apologize.
So
Annie White just told me who it was. So
it's kind
you
of on
might
the
even
slope
have to
here,
corral
and
it
you're not
into
gonna
the
be able to sit
box.
down
That's
in
why I'm
front of it if
suggesting
you
if
got
the
the drawer
box was
open.
open, if it starts to swim.
In some cases, I would agree here. The problem is if we have the drawer open, that makes it a lot more difficult to set down
Okay.
for a really stable grab.
Okay, yeah. all right,
Otherwise,
thank
I
you.
would certainly like to, and to get it in there as quickly as possible.
Okay. And also, Chris tried to grab it around the thicker base there, and then it just took off to the left, if you will. So, if you can, I'm wondering whether even if you could try to grab it head, you know what I mean, sideways, so you prevent it from swimming off.
We'll do our best here.
Yep.
We'll think about this before we try to grab it, but.
All right, video, come on back out, and we'll start to get set up here.
Okay, I'm just going to grab a quick specimen view.
Oh, sure.
Well, actually, at what point did it start to swim away? Was it when it first felt contact with the craft, or could it kind of feel it?
No,
when
it first felt the manipulator touch it.
Okay, copy.
Okay, full wide.
All right.
So what I'd recommend, actually, why don't you tell me what you're thinking, Dave?
Well, this is a nice place to sit down, but I have the rock in the way, so I'm thinking I go over to the left, turn almost 90 degrees to starboard, and possibly come down over its body like that. I'm not sure, because then it can't swim out, at least. What do you think?
Yeah, I'm not sure you need to go like 90 degrees from it, but kind of at an angle. Yeah, just when you're only going to be able to get the one toe in.
Yeah.
Looking at the event log.
Unless
Sorry,
you
I've
can
been
kinda
so
try to
focused on the
put your
video,
other
I haven't
starboard
noticed
toe on this
the
other side of
event
that
log.
rock.
Maybe.
But I don't know if bumping the rock would cause it.
Scott, we'll
startle
never
everything.
find one on a rock it's just a really big beast. I don't think it'll work like that, but you'll see
Ship
when
has
we
stopped
try to
connection.
grab it what it's going to do.
Yeah, it stopped 2-3 minutes ago.
Okay, perfect.
So yeah, you might have a minute or so swing, but besides that, you're good.
Good.
Yeah, I think Tina was not on, but Asako was, and Asako remembers exactly what it did, and it surprised the heck out of all of us.
Co-pilot, can you joy lock me? Oh, never mind.
So,
It's pretty far down. isn't it?
yeah. A lot less maneuverability with the arm if you're high up like that.
So go one step further. And back to the front. I think I have to go up there. Otherwise that angle's bad. Pull back and get a better look.
Yep. Just take your time. it's fine to experiment a little bit, try to find your best footing.
It's a difficult spot. So you think perching above it is?
Well, it just, you might be able to set your port toe down on the rock on the left side, you know, and the nice thing about this is you can just come in from the top, so you wouldn't have to reach down too far, but then, you know, the further above it you are, the less maneuverability you have, you know, to come on top of it, so. It's the fewer angles, a smaller angle at which you could grab it.
Right. But coming around that way is also a problem if
I don't,
further down.
Yeah, I'm not sure. I'm not sure if I'd approach it from that angle, because...
You think more head-on?
I think coming at it from the starboard side as much as possible, because even then it's, I mean, I understand your strategy with keeping it kind of boxed in, so we can't swim away, but, you know, if you're kind of angling the grippers, the claws toward the rock, you know, you can try to get it trapped there.
Okay, we'll try this.
I'm going to see if, well, we generally want to remain a little bit quiet because this is a difficult collection, and so that's why we're being real quiet here while they concentrate and discuss how to do this. And so as soon as we got this in the box, I'll relay some of the chat room comments.
Joy lock me?
Sure. Perfect.
Heading back.
So let's say you go for the grab here. What is your strategy with orienting the arm? This is important to think about before you go for it.
I think I have to go back past it and come back in with the wrist about that angle. Grab it around the base with the wrist facing toward it.
And what are you thinking? The grip force, and how are you gonna pick it off the rock, and where are you gonna put it?
It's gonna go port side. Probably 3 grip force. I will come in like this, grab it around the base, pull it out.
Okay. And how are you gonna deal with the grip?
What do you mean?
Are you just gonna grab it?
I think so. I think I want to do it fast. Right?
Okay. But do you wanna clinch it too tight?
No.
Right. So you're gonna try to feather it in.
Okay.
And I think it's worth, you know, before you get too close to it.
Yeah, do a couple
practice feathers and try to even come in. So you just have to get a little more and close it around the jaws.
Okay.
And then what's going to happen once you have it in the jaws? What are we going to need to do?
I'm going to need to lift off.
Yep. So you'll have to lift off, and what are the mechanisms through which you're going to do that?
We'll come out of joy lock, probably leave auto heading.
Okay.
And come up and back.
Okay.
Lock in depth.
Okay. Yeah, and with the auto depth, what I'd recommend, you get up off the bottom. You lock in auto depth, and then you give the set point a little bump up, such that the thrusters will keep trying to push you up, and you'll have a little bit of momentum. keep you up, instead of if you have momentum upward and then you lock it in, and then it has to fight to come back down, and that can be precarious. So, yeah, I think we have a plan. So, when you're ready to execute,
all right,
we
prep
can
starboard bio box.
Yep.
And spin up.
Starboard or port?
I'm sorry, port.
Okay.
Definitely port. And are we spun up?
Hydraulics are ready to go.
Great.
Grip force 3.
Do you want me to take your camera?
Sure.
Okay.
I need to take both, actually.
Okay. You want me to put up Mini Zeus?
Yes, please.
Okay. Sean, if you can help me keep an eye on the Sirius camera while we're distracted here.
Yeah, copy that. I'm on it.
So, bring. Not getting wrist rotation. Am I in continuous? No. Oh, I'm against the limit. Okay. Struggling a little bit with the wrist here. So co-pilot, I'm hitting. I want to have some range of motion on the wrist rotation.
Yeah. So when you put it in continuous, and you can go as far as you want. Yeah.
But then when I come back out,
you have to bring it back. It has limits in manual rotation. So if you're hitting a stop, you got to spin it, or just bring it back. Yeah, bring it back the other way until there you go, until you center it out.
Right, but it's less than one full rotation. Okay, there we go.
And watch how close you are to the Mini Zeus here with your wrist.
Can you come up on main?
Yep, coming up.
Still struggling with the rotation a little bit. So that's a stop.
Yep. So you can rotate it and then index out, bring it back, index in, keep rotating until you've centered out the rotation.
Okay, center the manual range.
Yes.
Go to continuous, get it about where I want.
Yes.
Then nope.
So don't even put it in continuous, just keep indexing in and out and rotating it manually until you've found the center of the rotation.
Right, that's one end. That's the other.
So watch your wrist. You should bring the arm down a little bit. You're really close to the Mini Zeus. Yeah.
That's the center.
Okay.
And then I want to use continuous to move the center to where I want it. And now I can't rotate left.
I'm not sure if the continuous is going to fix the set point. I always do it manually with indexing in and out. I don't even touch continuous when I hit the stop. I index in, I rotate it manually, index out, bring the controller back, index back in.
That's what I'm doing, and I can't go any further.
Oh, I'm not sure. That's something we'll have to look at later.
And I can go further. Okay, that's a stop, bring it back, can't go further. And I can go further with continuous. Alright, I will just deal with it. How does my alignment look, Copilot?
You can look in that manipulator and see that you're a little far forward of it.
Okay.
And just one more note I'm going to say before you go for it.
Yep.
It might get away, and that's okay. But just nice, slow, controlled movements. It starts to swim. Let it go, you know?
Okay.
I think you're still, if you look in your manipulator camera, it looks like you're pretty far forward of it still. Yeah. Watch your claws, your outside claws are about to hit it.
Well, there goes the stock. Got some tentacles.
So we're going to back up, come off. Auto depth, bumping it up.
I would go ahead and bump maybe auto XY as well. That'll keep you from going too far with the current.
Okay, can you open that box?
Yeah, you got auto altitude.
Whoops.
Yep. There we go. Drawer is opening. With port bio box open.
Great.
Tilting down on Mini Zeus. So once you get it positioned in the box, why don't you index out? And then give yourself a bump upward on the auto depth.
some control problems beyond the wrist.
That's fine. And just give yourself a tap up so that water gets forced down into the box. Looks good.
Okay. Ready?
Go ahead. Oh, it's stuck.
It's stuck.
It's alright. Nice and slow. Get it in the box. Everything's safe. Just nice controlled motions. You can try rotating your wrist, jiggling it back and forth. It's alright, nice and slow. You could try kind of lightly scraping it off on the side. But just make sure you don't put too much pressure on the box.
Okay.
You're almost there.
Can you see where it's stuck?
You could try rotating the wrist. I can't quite see. There you go. Oh, is that it?
That's
all
it.
right. Clear the arm. Arm is clear.
Arm is clear.
Retracting. Okay. Drawer retracted. Pins retracted.
Okay, can you follow me to stow?
You got it.
Actually, I'd like to run the arm through some movements. I was having a little trouble there. I just want to make sure everything's okay. Yeah, there's that stop.
Something I'll have to play with later.
Yep.
I'm not sure.
Right, they just, let me update Chris for a moment.
Okay, bring it up to stow.
So this is Chris Ma from the National Museum of Natural History. I'm back from lunch and ready to
Nice job,
ready
Dave.
and raring to go.
You got it.
So if we could head up at your convenience.
All right, taking my camera back.
All right. I'll give you Mini Zeus back as well. Sure. So I think we're facing 065 right now. I think 070 was the last move, and that looked pretty good
I'm
to me.
centering up.
Yeah, that's correct. 070.
Yep.
Last move.
So I'm looking there now.
Centering up.
kind of zigzag back and forth, and maybe we'll get a move in here pretty soon.
Yeah, and remember the lower port was stored.
That's right.
We could bring that back out.
We'll bring it back out. Thanks, Sean.
Check your tether right under you.
Yeah, hold on 1 second. Sure. I'm pretty high up, so I think we're pretty safe. All right.
Yep, it's fine.
Looks good. 18 meters above you, so.
Yep, I was just looking at this.
Good.
Wanted to make sure it was open.
Do we want to look at this brown thing
Yes, I'm
coming into
getting
the
set
center?
up on that. Let's turn lasers on.
You got it.
And you can come in.
Looks like a dead sponge skeleton. And what's this below it? There's a dark...
Low.
Yeah, there's a flat, dark structure that's just...
Oh.
Yeah, let's zoom in on that.
Like another 4 amp possibly, Chris.
Oh, is it a 4 amp? Ah, okay. Okay, we can get a... If we don't get a establishing shot, we can just move on.
Okay, video, please come back out. And we'll continue up.
Thank you, sir. Much appreciated.
I take that back. Snap in on this.
Nice feather star. Echinoderm filter feeding. Uses its arms to pluck organic particles out of the water. Undoubtedly one of the many that we'll see today. We're good. Thank you.
Okay.
Can we hold just 1 second?
Sure.
Looks like it's got some nice eggs or something on the pinnules near the mouth. Fecund crinoid.
Okay, video coming out.
All right.
Full specimen view.
The laser's a little closer.
Thank you very much.
Come on out. Now let's make a note to not open starboard port bio.
Note already made.
Great. Something in the water column? Let's
Yeah, I
try
see
and
it.
get that. Go.
Oh, neat. Looks like a little radiolarian or some kind of protist. Oh, wow. That's cool.
Nav, can you watch my swing on?
It looks like a satellite.
Yeah. Little Sputnik.
Let's let it go.
There's a holothurian up near the top, a little bit to the left of the screen.
What's that purple thing up ahead of us? Is that just a dead sponge stock, or?
I think it's about to take off. Let's come in.
What is it? Oh, it's a sea cucumber. Big sea cucumber, I'm guessing. What are the lasers on that?
Come out a little bit for lasers.
Yeah, they are.
Wow, it's 10 centimeters long. So this is some kind of a lasiopod sea cucumber, maybe in the Elpidiidae. Notice it's got the long tentacles coming off of it. The edge on the left side is where the head is located, and it's got its feeding tentacles, like it's prepared to, and you can see some of the sediment that it's ingesting. It will devour the goo that's sort of rested or floated down on a lot of the sediment, and it swallows it. It goes into the intestine, which you can see very conveniently through its transparent body wall. These then can essentially cycle the sediment through their body, and they deposit sort of the clean sediment cast afterwards out of the end. So sort of like an
Should be
earthworm.
stable. You can come in.
This one's quite large, so this is a productive lifestyle for some species. It's also got some interesting things growing on the
Let's go ahead and put a 20-meter
tentacles.
Copy.
I'm not sure if that's epizoic or if that's just part of the animal, but these are important for bottom ecosystems in the deep because they help cycle nutrients and other items, as well as clean the sediment up,
Go ahead now.
or aerate the sediment for different kinds of animals that live in the substrate. Great, we can move on. Thank you.
20 meters at 070.
Video just wants to
Chris,
stay
do
on
you
this a
know,
bit.
has anyone hypothesized what the function of those elongated podia would be? I mean, they're not like the sails of the swimming ones. I'm not sure what
That is a good question, Scott. I honestly don't know because, I mean, I think some people have thought that they might be sensory in some way.
ROV bridge, move of 20
maybe
meters,
to
bearing 070.
detect water current, or maybe even to detect how the goo that they like to eat is coming down. Dave Posson, the curator at the Smithsonian for sea cucumbers, has mentioned to me that you'll often see these, if you see them in a massive field, they're all oriented in the same direction, sort of directed towards where the food is coming from, and so possibly they're able
Chris,
to...
Sorry to interrupt.
Yeah.
I think there's a little white branching structure just near the head end of this thing that might be a fry zone just past it, so if you're still there...
Yeah, I think those were little forams and things that we were seeing earlier. Those little branched.
okay, I
yeah.
thought this was something larger.
Oh no, it's well. Let's pull out if we can.
Behind the hood right now.
Video, if you're happy, you can come out.
Is he talking about the white structures growing on the holothurian?
No, I don't think so. He's talking about something on the rock.
Find it on the rock, and I don't think it's a foram. I think it's a little bryozoan, and it's partially obstructed by the oral hood.
Okay.
But when we first zoomed in, you could see it very clearly. So we
For
can
wide-
look back at the video later.
Yeah, that's great. We can move on, pilot. Thank you.
Okay.
Nothing makes Wednesday better than a 10-centimeter long holothurian.
I can't think of anything.
Unless it's a 20-centimeter starfish eating that holothurian.
All right, so another 3 minutes or so, we'll start feeling a move upslope. But until then, if you kind of want to come to starboard and pan across.
Got a swimmer here. come in. Let's see if we can get this.
Blowing them
Switch to
around.
This one. All right, you can come back out.
Actually, yeah, it kind of feels like I'm seeing that move already, so.
All right.
Let's take a quick look at that branching structure over to the right on the boulder there. Looks like a toothbrush.
Oh, okay.
Thank you.
Right above the lasers?
Yeah.
Okay, come in.
I don't see it.
Right between the lasers now.
Yeah.
Oh, okay, I got it.
Coming down. Go.
Okay. Looks like a little tube worm of some kind. And let's pan left also. Our intrepid. We could move to the
Let
left.
me just get this. Okay, we got it.
Aravina Bridge, move complete.
When you're done, of course. Thank you.
How far left?
Looks like a
It was only a
polychaete
20 minute
sandworm
move,
with
so
some barnacles
you're
growing
fine.
on the tube,
Yeah.
perhaps.
Ah, here we go.
Oh, okay.
Oh, is that a? That's not a black coral, is it? Or is that a juvenile? Yeah, if we could zoom in on that. As
You can take some more.
so, if our shoreside talent could chime in on this, I think that's a. Is that a black coral?
Yes, well done, Chris Moss. That looks like maybe a very small Bassapathes black coral.
I must relinquish responsibility to Nicola, our intern, who is
Do you have any
far more
video?
sharpshooter of an eye
That's
than I
excellent.
did with this one.
There's something near the top of it.
Yeah.
The question is, what is that little orange thing that is on one of the branches?
That's a good question. Lots of those today. It might be, I don't know, a parasite of some kind?
I'd love to get closer, but this thing is tiny.
Yeah. If this is the I think we got enough of a shot of the front. We can move on. I think that's enough for them to see, at least for now. We might even see a bigger one later.
Okay, video whenever you're happy. And we'll hit the base of it with the lasers. If you come out a little more.
So, the name of our expedition is the Lima Okamoana, which means Partnership for the Ocean. And right now, there's
Maybe
nothing that
you range down
demonstrates
on
that
D2 sonar?
more with regards
So
to
it'll
our
be at 30
research
meters
than the collaborative
pilot.
efforts we have between people such as our junior scientists,
There you go.
such as Nicola Rodriguez, who helped spot that black coral, our pilots,
Hey, Sean,
our
let's,
videographers, and of course my
actually,
colleague,
right.
who
Why don't you come up,
is
We're
switching
gonna
out
do a
with
steeper
lunch with me
feature.
right now. I've already gone. He's already
Something
down there.
should I not
So
stop for it?
Yeah,
big efforts like
come
this
up.
don't happen by individuals. It's about a team.
I think you need to come up vertically. See, it's a
Oh,
little,
yep. Oh, wow.
little sharp here.
It's pretty steep.
Copy that. So we should have maybe a minute or two of swing left.
Let me just
Wow.
locate
Yeah.
the top of this.
Punch it up.
Yikes.
Then we can take a look at it.
Yeah, I was seeing over it, but didn't want you to get caught down there.
Sure.
So we're looking at a large magma boulder thing.
All right, that's the top.
Okay.
And we're coming up over the top.
So when
wow.
we can wait for Sirius to settle out, and you might be able to get back down there.
This, I believe, is where the crest, the ridge, or something, or rather the ridge begins.
So yeah, if you want to come to your port, I'm going to bring my head back to 070 or so.
Video switching out.
Thank you, Emily.
And Chris, if I recall, we believe we are on the lee side of this mini ridge that we're climbing, and so when we get to the top, perhaps the currents will be stronger. Is that correct?
I think so. Yeah. Where are we? We're right. Yeah, we're right on the contour line.
How much left in the move?
Move ended. I would bet
okay.
Sirius is done swinging.
Okay,
It's been almost 10 minutes. Exactly.
Yeah,
20 meters, yeah.
Yeah.
Well, I'm looking down at you. I'll keep the camera at like 80 degrees, and I'll keep an eye out. But I think you can explore a little bit if you want.
All right.
So what's the current like here? Is it?
There's very little right here. I think there's a little bit in my face, which is down the slope.
Okay.
Out of the east.
How you doing,
but
Roland?
not much.
Okay.
We have just
Doing very well.
minute or two here that I can poke my head back down the wall before we need to
if
get out of here.
you have a moment.
So we'll do
Some
that
of these
quickly.
little white. Oh, there's a coral or something like it. That branching structure right in the middle.
In on this video.
Yeah.
Ready when you are.
Come in.
Oh, this is our first coral of the day, I think. And I think it starts with a chryso. What do you think, Scott?
I would agree with you, Chris. I would dispute it's the first coral of the day if you include sea pens and black corals. But it's the first octocoral that lives on the hard substrate, so I'll agree with you there. And it is a member of the genus Chrysogorgia in the family Chrysogorgiidae, which are the golden corals, and it's very clear there. You can see that golden color through the thin tissue. And this is a really nice angle to see what Les Watling has said several times about how it is you characterize these. And we're getting to a view now where you're almost seeing 3 tunnels going down from the top of this colony and through it.
Oh, wow. Yeah, that's right. I can see that.
Branches, as it grows, is perfectly aligned with ones below it. And one of the ways we determine the taxonomy is how many revolutions of the main axis do you have to go to for that branch to be perfectly aligned over the lower one. And I wonder if this is Chrysogorgia tricollis, based on those 3 sort of openings that...
Oh, is that where the name comes from? Interesting. I noticed, too, all of the polyps are oriented in the same direction. That's always fascinating to me.
Yeah, I think that's just showing you the direction of the currents right now. They're pretty delicate polyps, and they can be bent over. It also looks like some of these polyps have the saddlebags that Les was referring to yesterday. So you see where the polyp is connected to the skeleton. Many of them are much brighter white, and that's basically gonadal material, probably eggs.
Oh, really?
And there's such a high production that the bottom of that polyp sort of extends on both sides over the skeleton, like the saddlebags on a saddle of a horse or on a bicycle.
Oh, neat. Okay, well, I consider myself edumacated. Those are reproductive.
Can you tell us anything about those 2 commensals on it? Or what a commensal is?
Yeah, absolutely. And so in the branches there, sorry, my video's frozen. Okay, sorry. So yes, in the branches of this Chrysogorgia, you see that there are 2 squat lobsters, and this is not unusual for many species of Chrysogorgia and other octocorals, and they probably have a pretty tight host association, meaning that this species of squat lobster may be very specific to living in just Chrysogorgia, for example. And it's entirely likely that we may have a male and a female. We have seen that on past expeditions when we've made collections, or maybe it's 2 females, both of whom have eggs, and they're basically probably using that colony as a perch and as protection. And so this is one of the reasons why we find exploring these deep-sea coral communities is valuable, because the animals themselves create habitat for other animals to associate. So it's kind of like birds living in the branches of trees in the forest. That's essentially what those shrimp are, what those squat lobsters are doing.
Great, thank you, Scott.
Sure is. Leave it to Roland. He'll get you what you need.
A little bit of fishing.
Well, there hasn't been much to see. I think that's the first associate we've seen.
Oh, there's another one up on the top there in that little sort of
straight above lasers.
Oh, yep, I see that.
Uneven area, yeah.
All right, video, come on quickly. I think it's the same.
Yeah, it's the same one. But here is another Chrysogorgia with its 2 squat lobster commensals living happily.
And so we were just talking about associates, so perhaps this is another good example. You don't see any of these squat lobsters on the rock around these corals. We're only seeing them on these corals. And we didn't see it on the black coral or the sea pens we saw earlier. So that's an indication that at least some of these, what we call associates, invertebrates that are living in association with a coral or a sponge, have a preference to live in certain kinds of species of coral or certain species of sponge.
Video's
and
clear.
it suggests that the larvae of these animals are able to detect using chemicals in the water that are given off by the coral, so they can detect them, land on them, and then perhaps grow there. Alternatively, as juveniles, perhaps they're moving around the rock until they encounter this colony and then move up to it and stay with it.
Continue down laterally
An
for a little
interesting
bit.
question, and one that requires further study about how long is this relationship in the lifetime, for example, of the coral or the crab. We certainly know that hopefully today we'll get to see some of the Metallogorgia octocorals. We know that the ophiuroid brittle star that lives with those stays with it its entire life.
Yes, and when we see these in the wild, or when we've seen them in the past few days, it's really strange to see how they always have an associate present. Very rarely do you see one without. Rarely do you see one without the matching set, as it were.
Right-hand side of the screen. I think it's an illusion.
I just see sediments and cracks. Yeah. All right, I'm going to come up over here, and we'll go on top.
There's some new guy at the top left.
Top there. Oh, maybe it's just a rock.
Are we running with lasers, or?
We had just turned him off for that associate shot.
All right, and we're up over that little wall.
Lovely. So we're tracking over what looks like a wondrous field of...
went off screen up left, maybe.
Cooled and solidified post-lava rock.
Sponge upper right.
I see that sponge. I'll go for that.
I'm going to sit down super close to it.
Oh, and there's a nice euplectellid over on the right there. It's a glass sponge coming up on the upper right-hand corner.
It's like a dead attachment point passing off the bottom of the screen now.
There's also a stick on the rock to the upper left.
Yep, I see that. I'm going to go for the sponge first.
There's a chance we can look down inside.
So these are animals made of glass, and we're seeing as we draw in, you'll see that their body structure is quite elaborate.
So I'm back in time for a sponge.
I had one specially made for you.
Thanks.
Get a little bit closer before I sit down.
You Plectellid, if I'm not mistaken.
Yep, you are not mistaken. It's just
Okay. Video, come on in.
So sponges are in many ways like the corals that we've been seeing. They provide habitats for a lot of different animals. In the case of some of these glass sponges, there are actually some fairly elaborate behavioral stories that are recorded around them. In Japan, there are some shallow water glass sponges that have shrimp that enter into them, into the internal chambers as juveniles. And when they mate, they stay together, trapped within the glass sponge cavity because they've become too big to leave.
Copilot, that's probably enough with lasers.
This is a euplectellid vase, as you said, and it could be one of two. Could be one of several actually. Could be a dictyolis. It looks more regadrella to me, so I think I'm going to guess that for the annotation log.
Regadrella?
Yeah, so these are sort of classic glass vase sponges, and this one's got what's called a sieve plate over the opening on top of the hole, the main opening, kind of like a bottle, where the water comes in the sides and then it's sent out through the top. And this is one of these sponges where I was mentioning the other day that they may be taking advantage of a Venturi effect. This morphology may actually be creating a Venturi effect where water passing over that opening draws water through the sponge at a more effective rate for filtering.
I always love the biophysics of filter feeding animals.
We're probably good on this one. Our video is good because this is a pretty common sponge.
Okay.
We haven't seen it today, though, have we?
Yeah, but it's recognizable, so thank you. And we'd like to get up near that crest if we could, please, and get into a really nice community with any luck.
All right. Should we get another 20-meter move in?
Let's do it.
Yeah, I see cameras at 5.5. Still want to go at 070?
That's fine.
We can adjust maybe
more to port. Yeah, that's what I was thinking.
Copy. How's
this is Bruce again.
6.5.
I noticed that
6.560.
we saw a couple of fish early on when we were in the
060.
sediment.
Copy. 20 meters. 060.
Into a rocky area. It's been a long time since we've seen any fish, so it'll be interesting to see what occurs later on.
Okay.
Yeah, thank you, Bruce. I think we're all kind of eager to see what the top of the Seamount looks like. After yesterday's find, we saw some stuff yesterday in the bottom, but when we got on top, that was the glory hole right there. So I don't know if this can be repeated on this dive. But it certainly got people curious.
5 degrees.
Yes, sir, that's correct.
All right, thank you. Coming up.
Yeah, I was just telling our the executive officer of the Okeanos Explorer that there's nothing quite like sort of having the camera roll up on a ridge or something, and then automatically having all of these sponges just in all of their brilliance just show up suddenly like that. The ensemble appearance is really quite stunning.
So we still have a lot of loose rocks here, too. I was expecting it to... I mean, we're fairly close to this little saddle, so it's not surprising, because rocks could have been moving down slope from the northwest, too.
Snap on it. Come in, video.
Dead sponge base. It's probably the base of one of those vase sponges too, because some of the euplectellid vases kind of have some finger-like structures.
No, it's like it's got a little
worm tube on it.
Yeah.
Yeah.
Pulling out.
Okay. Copilot lasers on, please.
You got it. Lasers on.
Because before I actually
Something under that rock to the right.
switch my switch on to active
hold
audio,
off,
I did
getting
sort
too
of
much
take a
further
quick
ahead of me
glance through
until,
C-Scribe to see what you guys had seen, and you saw a bunch of chrysogorges that looked like along this wall.
Oh yeah, there were some present on the vertical surface. Some Chrysogorgia tricollis, yes, with some squat lobsters. Scott gave us a very nice presentation on that.
Some white dots lower left. Is that an urchin on the top, or not an urchin, but an enemy?
Where?
Or just a
Now
shadow?
the swatch lead.
It's a rock or shadow. Stalk in the upper left and a whip behind it.
Yeah, I'm a little far away.
Something orange on the right-hand side of the rock.
Is it a shrimp?
Yeah, you're pretty far out ahead of me, Dave.
Okay.
The white dot on the right.
Let's look what's under this rock first.
Okay.
Come in.
It's another one of these mysterious mucus nets that we've been seeing. Oh, some sponge spicules left in there.
I don't know if there's sponge spicules. Maybe. I don't know. Some type of thing that got caught in the mesh.
Video's clear.
That's enough on this amount.
There was like a white disc on that. Anyway, that's a dead sponge stock, I guess. There's also a white sort of dot on the right side there.
All right. Come in for the dot.
Dead stock, I think.
And another stock. Freaking sponges in their stocks.
Got a real eye for those, Chris.
There used to be a lot of stuff here.
All right, co-pilot, you should start feeling serious
swinging
swinging
visual
now.
exercises of surveying a terrain like this is that amongst all of the dark background, we have these little sand pools, and they often have sort of star-shaped to round-shaped outlines. So when I and my video colleagues are looking for things that we might possibly visualize, we go immediately to that and then realize, nope, it's just geology fooling you.
Yeah. Sorry, I thought you said go for that.
I will do.
Yeah. Copy.
So I am stunned on how barren it's been actually of biology. It has been a little slow starting out.
Yeah. Let's get a quick zoom on this crinoid just to see which one it is. Thank you.
Come in.
Wow, what is going on with the Siri on this thing?
Well, looks the basal ones, the ones closer to the disc, look like they're reproductive. I think I saw something like that before.
Excellent.
But yeah, it's a strange looking one.
Yeah, the little sort of swellings is what you're saying, right?
Yeah.
But it extends onto the feet, the Siri, in your experience? Or?
Well, I mean, those are the ones that are, those are the pinnules that are closest to the cup, right?
I see.
So it doesn't extend past that. So
No, you're seeing some swelling. Are those the pinnules, or is that?
So
oh, you're right. Okay, sorry.
Yeah.
I was
No,
looking at it.
We can move on. That's great. Thank you.
ROV nav, move complete.
That's some video footage for our friend Chuck Messing, who frequently helps us with all things crinoid and feathery star.
Come back in.
Oh, wow, it was a nice little
is that a black
coral? Yeah, second one that I've seen today.
It looks like, wow.
Partial.
All right, shoreside coral people, here's your black coral.
I've got an idea as to what it is, but I'm hoping Tina is active in the chat room right now. She is, and she lost the video. Doggone it. Hopefully she'll get it back. Tina, a very long stalk, and then at the top it sort of has a single set of branches coming out in alternating fashion. Could be umbellapathes, it could be a long bathopathes of some kind, something like that. Okay, Sako's got it back, so then maybe Tina's got it back too. And she can get a look and help us out with this, and she specializes in these animals.
Let's go full screen.
Okay.
Very long stalk on this thing, which is kind of reminiscent of umbellapathes, with just a single plume coming out on one side. But let's see what Tina has to say.
Video's clear. Whatever science is.
We're good. Thank you.
Okay. So the current is pushing me sideways along this now. It's out of maybe the northwest.
Out of the northwest, okay.
It's fairly significant. A little lower, there wasn't much current at all.
You said it was current out of the northwest part?
Out of the northwest.
Okay. Just making a note in the log.
At this. Let's look at this squat lobster. I mean, it's a what do you call it? Blind
I want
lobster.
to sit down super close to this.
What's it called?
Oh, polychelidid?
Polychelidid, yeah.
I was punching stuff in. I didn't even see that guy up here. There's a nice find.
And around to the
Have we seen a polychelid so far this cruise?
Not on this cruise. I don't think I've seen one since Diva's cruise.
Right. So these are lobsters. These are what are considered living fossil lobsters, if you will. That's the way I like to think of them. I don't know how true that is, but...
Well, we're all living fossils, but
right.
Sure.
Okay, come on in.
But they're really unusual.
Lasers are clear.
Odd though, I thought they were burrowers. It's just sort of sitting there out on the open. It's interesting.
Please.
Sure.
We'll tilt down.
Is this something that you've seen before, Chris?
Oh yeah, quite a few times. It's Homerion asper, maybe. That's one of the species that we noted in Hawaii, but Homerion is one idea, and then honestly I don't know how many different species there are. I'll check and see if Mary updated any of their IDs on this.
When did that move stop now?
There is no lobster category here.
52, so it's been done for 6 minutes.
Okay.
What do you?
Yeah, so
Probably
you're
at the tail end here.
Yeah, I wouldn't think you're swinging anymore.
Where?
Okay.
Oh, okay. Well, it's not that family, but that's okay. You know what I'm going to do? I'm going to change the family. I'm going to cheat. Yep.
Okay, go full screen.
To answer your question, I just did a search, and apparently there are 38 species of these blind benthic lobsters, the Polychelidae.
So we can definitely go to species, right, Scott? Thank you very much for that.
Go ahead, Chris.
I love how the
medium shot.
claws on this one have that sort of ribbon ended TIF. Neat animal.
All right, so every image of this in the Okeanos Animal Guide that Mary Wickstein down in Texas A&M has reviewed, she's actually gone to Homerion asper, to that species. So at least we can put a question mark on it.
Okay, video's clear.
All right.
Science is clear.
Neat.
That brings our time to an end.
So.
Very happy to.
Watch as you're back in here.
Okay.
See some rocks behind you.
I see that.
Happy to see the bottom with you guys, even if it was for only 20 minutes.
So, as we roll around to noon here, Hawaii time, we're seeing our first row of engineers and pilots engaging in their scheduled switch out so that the team that's been with us for the last couple of hours can go get their lunch, and our refreshed team can come in and take over for them. So this is Okeanos Explorer.
Hey, Nav, does Fernando know
in the middle of an exploration of an
I don't know.
Node or
I'm not sure his
previously
schedule.
Seamount.
Oh, okay. I don't know.
At a depth of 2,275 meters. We've been climbing a ridge.
Pilot, I was just told we're getting onto that ridge that they wanted to get to.
And we've just seen a lot of interesting deep sea critters, including a blind lobster, a member of the Polychelidae, as well as some interesting.
Yeah, it's ship stopped. Yeah, we want to make some way uphill to find a more
as well as some black corals.
Populous area.
And, looks like we're
last move was 060,
and so we're
current 0 setting.
Moving forward.
Actually, I think that might be.
If we could stop and zoom in on the sea anemone real quick.
There we go. Okay, let's get in there. I'm gonna get a toe down here.
So what we're zooming in on is an example of how even dead animals still provide habitats for any number of other organisms. This is what appears to be a dead sponge stock from a glass sponge.
Clear to zoom.
And what's happened is that it's provided a home for several other organisms.
Partial.
This, for example, is a sea anemone. I believe it's a flytrap anemone, and these will settle on these stalks in order to take advantage of the water current. As you can sort of see from the way the tentacles are extended out into the water. They're predatory, so probably catching little crustaceans and so forth. But sometimes you can hide a little critter right around the mouth of something, and here we see a cute little polychaete worm. I believe that's a scale worm, and it's living right on the surface of this flytrap anemone. I've seen other examples of this where the thinking is that the worm actually takes advantage of the food that the sea anemone eats. The sea anemone doesn't really benefit a lot from the worm itself, as far as I know, but there are often examples where the benefit to both parties is unclear. As we spool down the stock, and there's another polychaete. We're seeing examples of brittle stars in the family Okeanos. These are frequently commensals or animals that live on stalks rising up above the bottom of the seafloor. Again, these are probably taking advantage of the water current as organic particles or other types of small food are brought by the ocean currents by them.
A little more.
And these are captured on the spines that the arms are covered with, and at some point the tube feet sort of wipe them off and move them to the mouth for food. Looks like a lot of these are less in feeding mode and more in holding on mode. Oh, and look at this. There's actually a little black coral right down at the bottom.
Chris, we were talking about associates earlier of the Chrysogorgia octocoral. So here's an example of associates associated with a sponge, in this case a dead sponge. But then we even had associates on the associates. So you were talking about the
back up
scale
to the lower worm.
polychaete worms that were on the anemone. And as you noted, that is not an unusual observation, which suggests that there may be a scale worm species that is preferentially on these anemones.
That's right, and every animal that you're seeing here is conceivably something that is also unknown to science, and so there's still yet a lot to learn from surveys of these kinds of relationships. So as long as we're here, Pilot, I understand that you've just all had a big change out?
We have. This is the after lunch bunch.
I'm delighted to have you all here. Perhaps you could introduce yourselves.
I'm Dave Wright, sitting in the pilot's chair at the moment with the Global Foundation for Ocean Exploration. We operate the ROVs for NOAA. To my left, I have
Fernando Aragon, sitting as navigator.
To my right,
Andy Lister, sitting as co-pilot.
And far right in video,
this is Roland Bryan, just off camera.
And Tara Smithy, clipping the video.
And this is Chris Ma from the Smithsonian's National Museum of Natural History. I'm the co-biology lead for this expedition. We're assisted by
Nicola Rodriguez from the University of Hawaii at Hilo.
And currently engaging with other expedition details is Chris Kelly from the University of Hawaii. So, and we're moving along
I'm
this
looking
ridge
over at 060. Is that going to be the
completely undiscovered and unexplored
matchup
Seamount.
with HIPAC for our next move?
This area was only just mapped this morning, and so
Your last move was 060. Right now, the bearing to the waypoint 2 is about 065. I don't know what, according to HIPAC.
And so
I mean, according to sonar, that looks like the slope. So
moving upslope.
Yeah, I mean, 065 would be.
I'm happy with that.
I'm happy with that too.
Okay, are you guys good with a 20 or 30 meter?
Oh, 30 at least.
30 is good, okay.
Yeah.
Coming
Maybe
up, 30 at 065.
We'll try to find some life down here.
Leapfrogged out front. I'll get
Bridge to
down
snap.
into it again.
This is the bridge. Go ahead.
We're ready for another move, which is going to be 30 meters, 065, 0.2.
You're pulling me just a little bit.
Range.
I see you coming on over there.
Bearing 065, speed 0.2 knots.
That is correct.
Coming up.
Swimming object at the top.
Yeah. Let's okay, zoom. And coming right just a smidge.
I once again stepped out for a moment. And did I miss anything, Chris, except for a polychaete worm?
We did a little bit of a survey on a flytrap anemone with some polychaetes and a little black coral, and but mostly we've just been, yeah. The front row had a moment to introduce themselves, and
ROV
we're just
nav,
sort of moving
move
along
initiated.
on course up the slope.
Great.
Copy, bridge. Thank you.
Oh, boy. Okay.
Can tilt down a little bit. Interesting the way those flares
why
come in.
this
It's like
Seamount
rays.
doesn't have a lot of things on it, and yesterday Seamount had a ton of stuff on it. That's what we're trying to figure out, because it's a little bit incomprehensible. I get it, there's not much current, but
well, since
we're not
there's,
exactly in a different ocean here, not that far away from the other Seamount. So
there's nothing fascinating to look at the moment, so I will just let go and let myself drift.
Nav, do you know what we need to average to get to final waypoint?
Stand by, co-pilot.
I would guesstimate the current out of 330. And somewhere between half, 3/4 of a knot, maybe. That's totally a guess.
let's see what we got. Let me get caught up on the chat here.
Stick coming upper right of lasers.
Okay, they're talking about
like
you know, we saw a little bit earlier on.
So the time, the average time would be 0.04 knots.
So we've got
is that going through waypoint 2 or just straight to waypoint 3?
Here, and it looks volcanic,
straight,
I
but
guess.
it's pretty much the
Yeah,
same.
I'd say so.
There's one dead sponge stock over there, but nothing else.
Yeah.
And you don't see a lot of the skeletons. You see an occasional skeleton or here, but there are skeletons all over the place.
Yeah, there's a dead one up there. Yeah.
So kind of, I look at the skeletons when we get down, and if there are a lot of skeletons on the ground already, then it's good. It's a good indication that there's something upslope.
But wait.
But there just isn't here. Nothing's found this
saw
place.
one
What is that?
10
Is
minutes
that
before
one?
you came
It's a
through.
crinoid.
Is it crinoid?
To the
Oh,
left there, right?
Zoom, please.
Yeah.
is it just on the rock? I mean, up above. Oh, okay, we're getting this, but there's something on the rock top center, too, after this.
Okay, let's come out a little bit, Rolly.
I think that's another crinoid, actually.
Yeah, but it could be that weird purple one. There it is. If we can go top center.
Where it is.
What is that? Or is
it
that
back
just
in?
a... It's an optical illusion. I'm sorry, it's an optical illusion. There's not... That's not sleepy eyes, is it?
There's a busted...
Oh, what happens to the best of us, even if we're completely refreshed. I mean, there's something up there that looks like a sponge, but I think it's just a saddle of
snap
sediment.
on this just for ID.
Yeah. There's another little
one?
crinoid. At least the crinoids have found this place.
What?
Yeah. Well, it's always a... It doesn't take much to filter feed, presumably, so it's always a good place for it if there's current.
Yeah. So what is, what is wrong with this place? And what's so right about yesterday's place?
I'm gonna
Maybe there's a whole bunch of possible explanations. It could just be young. Could be there's too much current preventing anything from settling.
Just go max zoom and just see if there's anything living amongst the sand grains here.
Or, as Dave may illustrate, it's all here, it's just very small.
A second, I'm gonna get myself steadied up.
What are we looking at? I wasn't paying attention to the pilot.
I believe this is Dave's
sediment
close-up.
Yeah, small world. Now something is aggregated this here. There's a couple of little lumps as if they've been stuck onto one of those small things.
That's grasping, Dave. That's grasping.
Well, sure.
I know. We're all kind of grasping. I just asked for a rock zoom on a rock that looked like a crinoid, right?
Oh, here we go. Here's something here on the right. What is that?
That's probably a foram, the test of one of those amoebas.
Okay.
Looks like it has a pteropod shell stuck on it.
Well,
it's a small mollusk.
I'm not a scientist. I can engage in wild speculation without embarrassment, and then you guys can correct me.
There's something on that ledge. Could be a sponge, believe it or not.
ROV nav, move complete.
Copy, bridge. The move was just complete. Do you guys want to keep moving?
Yes.
Copy.
Yes.
Same bearing, same
Yes.
range.
Yes, yes, yes.
30 meters, 065.
Copy.
Yep. Let's do it.
Bridge, this is enough.
This is the bridge.
Let's get the same move in. That's going to be range 30 meters, bearing 065, speed 0.2.
Oh, that's interesting. There's a little
That's
hydrozoa
range
or something?
30.
I don't know what it is.
Bearing
I'm studying it right
065.
now.
Speed 0.2.
That is correct, bridge. Thank you.
Alrighty then.
We got any more zoom rolling?
That is max.
Max.
Bruce Mundy was asking for the quad screen view be put back on camera 3. You guys have got
What's
a
up?
chance? Great, thank you.
What else you got?
Let's see here.
RV nav bridge. Move initiated.
Copy, bridge.
Well, I'm not sure there's going to be a lot of wows today, Chris. I don't know. It's still early. Still got time, but...
We'll see. We saw that the polychelid lobster.
Yeah, and we did see the Sariantid that we collected. That was
Yeah, we
sort
can
of a
come
wow, but
wide.
we wowed that the other day when we first seed it.
Those are
So it
bigger
wasn't like
fuzzy.
a new wow.
See those?
That's kind of a secondary wow.
Make a little jump here.
Well,
So
I
white
live in
thing
hope
up ahead.
we'll make best of it, and I'm sure we'll see something good. Besides, there's always one thing I have noticed is that the sea stars always come out right at the end, like a good movie.
So, where are we with respect to this ridge? I mean, I understand we've climbed up the sort of western flank of this ridge feature, and now we have turned, I think, and are moving along the axis, but are we in the center near the edge, or is it actually too wide to have a clearly defined center?
Nav, do you want to
Thorian,
take
right?
that one, or would you like me to respond?
Come again, video?
Let's go look at this.
Sorry,
sea
I
cucumber.
can respond to his
that was watch
question
lead.
there. So Scott, we are kind of right, according to the bathymetry, we're right at the beginning of the higher point of the crest, where we're sort of in the saddle on the crest. So we come up
here and then
from
we'll
the
go for
sediment up
holothurian.
the back western slope, and now we're
Okay,
on the
wide,
crest,
please.
and we're in the process of turning to the north a bit to run up along toward the summit of this, and we're probably right dead center of the crest, and I think they're moving slightly to the eastern side, and they're going to try to climb up a little bit along the eastern side. But this should have good current.
Yeah, I just opened camera 3, Chris, so I could have a look. So the reason I asked that is I'm wondering if maybe you're being negative because we're still a little bit on the lower, flatter part of this ridge. So perhaps as we move up, things might improve. I'm gonna stay optimistic.
Yeah, so Scott, I'm going to try to neutralize your optimism, because you know I believe in the law of negativity, which basically states that if you expect the worst, good things or the best happen, and if you expect the best, worst things happen. And so when I express negativity, I am trying to ensure a positive.
But you're fundamentally optimistic, so
Right.
that's a fantastically bizarre set of magic you're working there, Chris.
After
That's
this
fine.
next move, Dave, we might
That's
want
how
to
it
look
works.
north.
Magic
because
has its rules.
I have a feeling it might start going
Thank you,
north.
Dave.
What do you think?
So this is
I
a
guess
sea
you'll see when
cucumber.
you get
This
up
looks
there.
like Hansenothuria. You can
Video's
see the
clear
clear
whenever.
gut, and one of my favorite things to look at. I never get tired of watching an animal digest its lunch, and we get to see it poop. So the day is young, but we can move on. Thank you, Dave.
All right, wide, please.
So these things are really common down here, aren't they? That's the most common sea cucumber. I mean, this species for the Johnston Monument in general.
It does seem to be present through. It's one of our consistently encountered
Fuzzy
animals.
on the right.
Yeah.
And that's always
Gain
fascinating.
up, roll in.
I'd have to look back, but I almost think we've seen one every dive.
Oh, I'd say that's right.
Yeah. All right, I'm going to run over to the edge of this ridge here.
So Scott, you're getting a good look
How
from
far are we in that move,
Dave
Nav?
Wright now, the pilot,
Well,
who's going to give
we're
you a look on
almost
the eastern
at the
side
end
of
of
the
the
ridge.
second move, and
Okay.
I'm pretty
Great.
sure
You can see from this view
you're
that
finishing
it's
the
just
first one, so
kind of a barren ridge, actually.
Okay, that's the east side. I'm going to pop over to the west side. So the axis.
Yeah, this is interesting. You know, back in 2015, I know you will recall that we had selected several.
I might go a little bit past it, but we can move me back, I
topographic
guess.
standpoint, geos that had these smaller cones on the top of the flatter tops, and we were hoping that those.
yeah, according to HIPAC, the
cones,
slope
their
starts to be
and the
about
fact that they were relatively shallow
340,
compared to the whole feature
350.
would be great places to accelerate current, and so find
And you said we're done with that
high-density
move?
communities. And we're diving on
we're pretty
something
much done
like
with the
that
second
today,
one, but you, I don't
somewhat
think you
different
have
shape,
felt
but
it yet.
essentially the same concept. And yeah, this is what we saw a lot. We were
Arvin
often
Nav Bridge,
disappointed
move
that we weren't
Copper
finding
Bridge.
the higher density communities. I think we did on one or two occasions find moderate densities, but nothing like the beautiful locations we were seeing yesterday and earlier this week.
So from where I
So it's
sit.
one of these cases where just when you think you're starting to get a handle on something, then you have a setback caused by a fact.
It's due north from me.
You know,
Okay.
it's
We can. I can
it's
turn
always
towards you,
wonderful
and we
to
can kind of move me
speculate that
back
all of
to
these
take
ridges,
some
and
of
this
that
is the
extra
same
swing out.
orientation.
Sure.
This ridge,
Okay.
you're pretty close to it. It's a smaller type ridge feature, but it's kind of the same orientation as yesterday.
So
And
now we're going to try to minimize
thinking we're
that
getting a
overswing
handle on
there
it, and then
for this next move.
Okay.
we get this.
So you want the next move to
But
be
the day is young. We are
a little
moving
bit
our way
west
up
of north?
slope.
Yes, I would think so.
And
Because he's facing.
we got plenty of
almost north,
chances
and I'm-
to see
Yeah.
a glorious community.
Yes.
I live in hope.
Yes.
There you
So
go. Now you're
350
sounding more
would be okay.
optimistic. I like that. What we need to find is
Yeah,
more
I'd be okay with that.
micro topography, some very pronounced knolls and pillows that are sticking out that might locally accelerate the currents. Maybe that would help.
Yeah, good point, Scott. Water's got to make its way around these kind of barriers, and that's the thing that we try to cue on. And
This
a
is
lot of the
bridge,
water
right?
in deep water, I think. Go ahead, Scott.
Range 20 meters.
Sorry to talk over you. We may have just passed another one of those stock whatevers that we were trying to
Range
image before
20
that was
meters,
flowing so much
bearing
in that
350,
strong current was
speed
very,
0.2.
very small, but we're too
Coming
far
up.
past it to look back.
So this looks like a
So
dead
stock
sponge stock,
being
Chris?
kind
Coming
of
up
lower
on?
center.
I think so.
To the right.
It looks like it's dead.
Just going off the bottom.
There might be some brittle stars on it, so it may be worth
Yeah.
a look.
Another one of those cucumbers down to the right.
Oh yeah.
Dave is right now kind of like a shark confronted with a school of fish trying to decide what target to pick. So it's amazing he was able to spot the sponge stock. Was that a little
we have move
sarcastic
initiated.
or what? No, that was a negative again. Sorry. Okay, I'm being optimistic.
Okay, we can zoom here. Roll.
I think it was the only game in town.
Off to the right edge
At
may have been a
least right
stock
here.
crinoid.
Yeah. Okay. I'm just going for the low hanging fruit.
Yeah,
here.
so this is sort of similar to the thing that we saw a little earlier. The stock of the sponge is gone for whatever reason, and a couple of brittle stars have availed
Co-pilot, you
themselves
have to be
of,
aware of the tether.
its
I think you're
body
moving.
to
Should be
perch
moving away from him.
take advantage
Thank you.
of the water current so they can feed, presumably.
Video's clear.
So I've got a potentially stupid question, Chris.
Sure.
Have we ever seen a brittle star bail from a dead sponge stock like we have from the corals? And by bailing, they just let go and they drop down? Have we ever seen that?
I don't know. I don't. That's a good point. I don't know that we have. These might be engaged in something else because it looks like they're coiled quite tightly. Usually when we see them on corals, they're kind of moving around through the branches. It also could be that these are separate species. So it's hard to say. But yeah, I mean, it's an interesting hypothesis.
Zooming on its
You
own.
know, do they are they a little more active when you see them on corals, and what's the difference between this and something alive? So what's the dynamic?
Chris, I'll say that it's an interesting question. I don't believe I've ever seen them bailing off of the sponge stock.
Yeah, and that's what came up about it. I don't remember him ever bailing off a sponge stock either.
Near the left leg.
It's always the corals for some reason. And now I'll kind of use it. Well, let's hear about the sea cucumber first. Sorry.
No, that's fine. This one looks a little different from the Hansenothuria that we were looking at earlier.
When did
this
that
one
move
has
at 60
more sort of
degrees
tube
end
feet
now?
or suckers
Should have
on
been
the
there
surface.
5 minutes ago.
But again,
5 minutes
translucent body wall.
Yep.
The mouth
Okay,
is
that
on
should
the
be the
left
end of my
side.
swing that way.
The
Little
anus is
center
on
up.
the right, and it's essentially photobombed by a little swimming polychaete worm. And essentially they devour the ooze that comes down and works its way into the sediment, and you can see right through its body wall, so you can tell what it's been eating and where it's going. They take advantage of the nutritious material, the organic material from the sediment, and that actually transforms the basis of the sediment after it's discarded by the animal. So these play an ecological role, sort of like earthworms do in terrestrial ecosystems.
I think this is a synallactid, Chris. Would you agree or not? I mean, they're transparent like synallactids.
It looks similar. The suckers are throwing me, though, so...
Yeah. That's a tough one.
Yeah. Oh, is that a stock crinoid on the horizon?
Could there be something?
I think the one I saw
could
is
be.
just left.
We're holding our breath.
Oh, no, it's one of Tina's things.
It's another one of these black corals, is that right?
I think so. Yeah, it's got that kind of stock crinoidish appearance. Oh, no, it's not. It's
a
totally
chrysogorgid.
Huh, weird.
Wait.
Yeah, it is.
RV-nav, move completed.
Boy, that's a young
Copy,
one,
bridge.
I guess. This be an Iridogorgia, a really small Iridogorgia, Scott?
Yep. Excuse me. Yes, that's exactly what it is. A small Iridogorgia, and that bodes well for continuing upslope. I see that it's missing some tissue from the lower side of the skeleton, and a couple of branches have broken off, and so I still think it's a young one. I don't think it's an old one that is falling apart because of the width, the diameter of the skeleton. And it seems to have a very big swoop or curvature to the spiral of the axis, so it's possible that this is a young Iridogorgia magnaspiralis, and that species, as the name suggests, is hallmarked by the wide spiral.
Snap soon.
Yeah, thank you, Scott. And now those are really tightly wound, woundly tined around that sponge stock.
Yeah.
And wide, please.
It did look on the view just before we were coming in here, is that on the downslope side, which I believe is the main slope facing the current, it looked like there were a lot more stalks, or several stalks. So who knows?
All right, co-pilot, the last move just stopped.
Well,
I'm gonna
something's
call
showing
another
up.
30-meter move.
Not
Let's
exactly
give me a
healthy
moment
yet,
just to make sure the
except for
swing's
the
done this way.
brittle but
Copy.
But I think we might want to go more at 340 rather than 350.
I'm looking
Well,
3.
sea cucumbers
Possible
would be fine.
starfish.
330 even, yeah.
Possible starfish center screen.
Okay, Rolly, talk me in.
Don't check. It's just dust.
Chris, there's a lot of rock rubble here. Do you already have 2 rocks? Or
yeah, you took the words.
wait until you get higher
Sponge
up?
on that little rock.
Yeah, you
just
took the
below
words right
lasers.
out of my mouth. I know that Dave never likes to deploy the manipulator ever, and he's on It's a pilot right now, but I wonder if we could twist his arm to start looking for possible second rock, since there are a lot of loose ones here.
Sure. We'll just rock on down here.
I said that because when I got on the ship, Dave's first comment was, you know, whatever you got to do with that manipulator, I'm there. If you got
There's
anything.
one up ahead with his round sponge on it.
I think everybody, all of the pilots are the same way. It's just. Where is that? You mean the dead one? Off screen right. Well, yeah, if you see something on the right side.
Oh, off there? In
Where,
the distance?
Where's he talking? I still don't see.
You mean on the upper right?
Upper right now.
Okay, here we go.
We're trying to find a rock that might have
lasers on to help you
creature on
locate,
it
or?
that might give a.
Oh, rock with creature? Well, that's
Yeah.
what Rolly's
no,
offering us here. Let's go look.
Yeah.
There's two different kinds. There's a.
No, the main thing, as you
Dent
know, Dave, the criteria here is angular with some girth, looking very volcanic-ish. That's our criteria for a rock.
That looks like the whole world in front of me.
There's something orange down there. I think that maybe it looks a little.
Oh, I was going
flat.
for the lower right.
what's
but
this?
Yeah, the lower right.
There's
That's
like
what I was,
of them.
There's a ball on the lower
we
right.
could do a
Yeah,
20
that's
meter
what
at
we were looking at.
330 as soon as pilot's happy with that.
Might as well zoom in on it. It's something alive.
Come on in, Roland. Let's see what this thing is here.
You said
Oh,
20
okay.
meters?
Yeah, we haven't
20,
seen this
yeah.
for a while.
330.
This is great.
But let's check with pilot
This
first.
is a a hyalonematid sponge.
Also note
It's
the
closely related to the polyopodon,
the
big
holding
massive
things
sponges,
go
but it's in
below
a
it.
different family. It's got the Lophophytus attachment to it. And we collected one of these sponges, two of them actually, in last year, I think. And it's called Hyalonema subgenus Corinonema. And these are a treat. I don't know why I'm infatuated with these sponges, but I really am. They're really weird looking. It's a cool family.
Well, it looks like a bunch of fiber optic cable wrapped up in a balloon.
But I think this is the first one of these things that we've seen on this cruise.
Yeah, I think so. I don't think I would remember something that looked like this.
Tilt
so
straight
they get
down to
quite
see what the other
large.
fibers are.
And yeah, they're really distinctive. When you have them in the lab, you can very quickly discern whether or not they're in that family by their spicules. They have very conspicuous microscopic parts or spicules that are called amphidiscs. And amphidiscs are the actual origin of the name for the order Amphidiscophora, because all of the members of that have this very distinctive type of spicule. So, and the other one is actually, could be even the same thing further up, but let's see if we can get a nice chunk of pillow lava here.
Yeah.
Okay.
There's another sponge on the left there.
Yeah, and it looks like it's just a larger version. They can get a little more translucent as they get larger.
If I call in a 15-meter move right now in that direction, I think we should be fine. We shouldn't go over him.
Unless they're already on.
I'd rather just wait a moment here. Nav,
please.
You have hydraulics are ramped up?
Yeah.
So interesting.
They start to develop a little more of an osculum on the top.
Little right, please.
But again, it's that attachment that's the real giveaway. You see how it sort of flares out like a broom or something on the bottom.
Huh.
It's got some weird multi-oscula on top, which is kind of strange. But definitely hyalonematid. Yeah, it's got a little different arrangement up there. I don't know what to make of that, but
it's weird that the holdfast
please.
looks like it's dead before the actual body.
Yeah, I don't think there's any tissue on those spicules, and so they tend to collect.
Something
I mean,
else. just below that?
Did I say that right? There isn't any tissue on those modified spicules called basalia, and that's why they can collect sediment and things.
So the previous pilot alerted me to some funniness in the arm, so I'm gonna do a little warm up here and make sure everything.
Co-pilot, you're not planning on opening any bio boxes, right?
No, I'm not.
That all seems happy for the moment.
It's a little flat.
A little flat? Okay. How about...
Can we get the lasers on, Dave, so we can get an idea
Lasers coming on.
how big they are?
Co-pilot is...
lasers are on.
That one's flat too.
Maybe just below the left of the dead spot.
How about this guy here?
Nav, how long ago did that last ship move end?
That's...
6.
Okay.
Should have been about 9 or 10 minutes ago.
It actually ended 9 minutes ago?
It should have, yeah.
Okay, well, I got 9 minutes of layback then.
It's too flat.
Everything here looks...
Yeah, maybe we can just move along, and if we see one that's got some girth, we can...
Just bump up the hill just a little bit here. We can leave the hydraulics spun up there.
Sure.
Co-pilot. And I'll just keep the arm handy here.
What is that? It's just a rock. Okay.
Here's a big fella.
Yeah,
up
that's. Is that too big?
I don't know. It's.
Well, that is. That would be great. If that were possible. It's meaty.
That's a big one.
We always endeavor to greatness here. Let's just.
It might be 2 of them, or is that all just one?
What's the state of our starboard rock box there, co-pilot? It looks to be empty.
That's a Nav question. Nav, do we have any rocks in?
We have a port forward. That's it.
Okay, then our starboard forward is free.
Is that about 50 centimeters?
60. It's, you know, I just like to get a hold of it and see what it looks like.
Don't forget, we have to get off the bottom too.
Well, we have a drop weight.
What if
co-pilot just be advised that in the port inner bio box there's a floater, so if we don't open it, that'd be the best.
I will plan to not open it.
So, boy, it is big. Just curious here. I'm not promising to bring it home. I'm just interested in seeing if it's even doable.
Hey, if you grab it, you buy it, pilot.
Jelly underneath just came out
from underneath it. Is it one of those hydrozoans?
Oh,
came out?
Maybe one of those jellyfish that live on the underside of the rock.
And he just took off.
It's the first time I've had one so heavy it came out of the jaws.
So who gets the workout of taking this out of the box and hauling it into the lab?
This is probably a good opportunity to introduce our intern, or our data manager, Matt.
Hey Bruce, this is Matt. Yeah, I usually get to carry most of the rocks. And today Chris and I both did a rock box restructuring, so we took all the rocks to the top deck. So that was very entertaining and a good workout as well.
Yeah, I had to change shirts after that.
Yes, fortunately,
once the ship comes,
this one might be too heavy.
Yep, this is.
Once the ship comes back to the dock, we cheat. We use a forklift.
All right,
well, I wanted to find out. Just whoops, wrong button.
Yeah, let's see if we can go for one that's we spot from a distance is a little more reasonable. That would be. I think we're going to find a lot of loose ones here. So
there's some other stuff right nearby here.
Chris, I was wondering if the side of that rock just broke off on the last attempt to pick it up and hidden on the right side.
Now here we have angular.
Yeah. How about, is that too long? This angular one that's to the left near the.
I was looking at the one right under the laser simply because it's
that's
the squarest of them all.
That's a good one.
We have many to choose from here, so we can.
Let's try the one you got your lasers on. That actually looks like a broken pillow segment. Unless John differs. I know John's on. He usually watches in the background in his office.
I'm on and dialed in.
Oh, good. So John, what do you think about this one? He's about to grab it.
Is that the one you were messing around with?
No, that's too big, John. This one's a bit smaller, but it's got a lot of nice angles on it.
Yeah, let me see when he picks it up.
Zoom, please, Roland. Let me get it up in the light here. Tilting up. There we go.
Yeah, not bad. It's a little small, but...
Well, let me show him the rest of the selection here because there's several right in front. Wide, please. So let me just set this aside.
I know my sights are a little bit big, but I was actually looking at that sort of elongate, longer one over to the left.
You were probably looking at this one?
A little bit further over.
Oh, that guy.
That one, yeah, because it's got a really nice flat face on it.
Yeah, that wedge-shaped one?
Yeah, that's what we're going to try to go for here.
Might be just out of reach. Let's see if I can.
Yeah, look for it. I like that one better.
Drag it over to me.
Turning out to be a little bigger than I thought, but he's got it. A little flat, John. I don't know.
Tilt me up a little bit, please, co-pilot.
Copy that. Taking camera.
I think it's too flat, Dave.
Okay. Wide, please, video.
Yeah.
So let me try this. And tilt me back down.
Tilting down. You want to try that one that's right there?
Yeah.
This one right here. It's a the deal about rocks is that if you get a good one, it's extremely valuable. The tricky part is finding a good ones when they're what you're really interested in is hidden underneath layers of manganese crust.
Well, now we can see
that one looks pretty good, I think.
So we have two finalists here. We have the original squarish one and the one in the gripper, or we can poke around some more.
Let's take this one in the gripper, I think.
Okay.
John, what do you think about that one?
Yeah, I was actually looking at that one before the other one as it was laying there. It looked pretty good.
Let's bring the starboard wing in.
Looks pretty good.
Oops, I'm taking your camera instead of mine. Yep, bring a starboard wing in.
Yeah, I think so.
Inboard, coming in.
Okay, we have a keeper. We could take a look at what those white things are.
That's just fragments of the rock behind it. Okay, that kind of looks a little carbonatey there.
Yeah, let's go with it. I think that'll work for us. Thank you.
Okay, so it's all the way inward. If you would like mini Zeus over there, let me know.
Just need a little reset here.
Perfect. Thank you very much.
Going outboard.
Had a question here from somebody. How large are the boxes that you use to collect the rocks? What are the dimensions on those?
Boy, that's a good question. I don't know because I'm an electrical guy, and I'm looking up and down the row, and we have skilled navigators, video people, and an electronics person, and all the mechanical guys are absent at the moment. But if somebody's listening from the workshop and could give us the dimensions of the rock box, that'd be nice.
So, Bruce, this is just Chris speaking as one of the people who's occasionally hauled a box out of the rock boxes. Roughly speaking, they're about the size of a milk crate, like a plastic milk crate.
Put the minis.
That's very inaccurate, but they're reasonably sized enough to accommodate.
And so we can see the rock in the box and roll
20 centimeter by 5 centimeter wide rocks.
So you can fit a 5-gallon bucket in one.
There we go.
So I know that.
There's one with a lid and there's one without a lid, and the one without a lid, they sometimes put in oversized rocks that aren't going to go anywhere when they surface, that are heavy enough and large enough, and probably smaller types of rocks, they've got a lid if they need to go there, and that'll make sure the rock doesn't come out during recovery. But I'm going to tell you one thing, that the boxes on the Okeanos Explorer are probably some of the best engineered collecting boxes I've certainly ever experienced. They are a work of engineering marvel. When the ROV comes up and we go down to collect the biological specimens, you can barely stand putting your hands in the water to remove the specimens because it is so cold. It maintains the cold temperature.
Yes, right? No, okay.
So yeah, it's
90 degrees.
It's really nice. They're very heavy walled, and the animals always come up in really good condition. What is that? Is that a Chrysogorgia over to the right?
Working on it.
I think so,
I
right?
can turn that way.
I don't know. It doesn't spiral. It looks more.
Oh, it's Anapathes, or excuse me, a Benthopathia.
Yeah, a black coral.
One of them. Yeah.
I think we're on the back side of the polyps. I think. There's also something possibly translucent, just downslope from it. Another type of coral. Are lasers still on?
They are. Swing them over there. Oops. Hang on a second. Got one toe down. There you go.
Translucent coral.
Clear to zoom. Coming left.
It's the bridge.
We're going upslope. I wanted to change the base of plan. Let's make it now 250.
I would like to see what Tina has to say, but I'd be
changing
more
bail to
inclined to say this
250.
is closer to a Bathypathes type than a Parantipathes.
Yeah, that would be my guess too, just from interacting with both you and Tina over the years. The thing is, I'm not sure Tina is on right now because it's pretty late in Moscow.
Changing bail to
She's
150.
showing up in the event log because I think she wants to get the event log chat after she wakes up.
I think we should put Bathypathes. These have very large polyps relative to what I think you would see on Parantipathes, and the branches just aren't quite right. So I think Bathypathes is the way to go.
Thanks very much, Scott.
Oh, there's a nice foram on the lower left side too. So this big leaf-shaped structure just to the lower side of it, that's another sort of sediment structure created by a single-celled organism. We've been seeing them all day today, but they're still very impressive when you consider
full screen coral.
What it was made by. Yeah, we can move on. Thanks.
Lasers off.
Lasers coming off.
And I didn't quite see what Roland was talking about there. He saw another coral somewhere nearby.
It's about 1.5 meters to the starboard side. Translucent.
Translucent. The Chrysogorgia, you think, Roland? Or
sorry, I missed those names. I think you might need to back up a little bit, Dave. It should be
soon. You said left, didn't you?
No.
Oh, I see it.
Starboard
it's
side.
Starboard right now, going off, almost off frame. I see it. It's going to be a Chrysogorgia, I think.
Left edge of the
left
screen.
now.
I don't see it. Oh, way out there?
Nope.
No.
Oh.
Well, we can probably move on. I think we imaged a bunch of Chrysogorgias already. We'll see if we can again get into a little bit richer hunting ground.
It's forward of us.
We are currently at 2,240 meters approximately.
Okay, we need a quick long distance look here, please, Roland.
And have we done the where we are and who we are a bit for a while or not?
We did when we came in.
The last time we did was when the shift changed in the front.
Okay.
So.
We are the NOAA ship Okeanos Explorer, and we are now inside the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument. Before I go on, I thought somebody might have come on the teleconference. Maybe they saw something they wanted to look at. Not quite yet. Okay, so we are in the Pacific Remote Islands Marine National Monument. It is a U.S. marine monument that encompasses a variety of atolls and islands scattered throughout the Central and West Pacific, as well as the South Pacific.
All right, shall we find our
Johnson
next
Atoll, where we are,
path?
or the areas and waters
like
around
due north
Johnson
from
Atoll,
me.
out 200
right.
miles. It also
I'm on
includes
board with you.
Palmyra
Okay, here we
and
go.
Kingman, a little bit further south, Jarvis Island, Howland and Baker, and Wake Island, which is much further to the west. So it's a really incredible monument.
All right, Nav, I am ready.
Got a
Pilot
lot
is
of seabirds, shallow water reefs, and we're helping them to understand what their deep water resources are
30
as
meters,
well
000.
in this 3-year
Sounds
effort
good to me.
here in the
Okay, coming
Pacific called Capstone. Our particular cruise has been named La Lima Okamoana,
Fish.
which means
Some
partnership in the
fish.
sea. We have a lot of partners, as you've heard us mention. And I'm
Oh.
going to stop. There's a fish.
This is Bruce, are you with us?
Let's get another move in. That's going to be 30.
Oh, yes, I am. So this is another one of the rat tail, or grenadiers, in the family of
That's
Macruridae.
range 30.
Same order
bearing
as the
000.
codfish.
Speed
this might be the same species that we saw earlier, but it looks a little different. It looks like it has a shorter intestine, rounder intestine than the previous one, and it's darker color. This resembles something that we've been calling the genus Kumba, which seems to be a relatively abundant genus at these depths, small species. You can see this is a fairly active fish, swimming rapidly, fairly good metabolism. They're active little predators, crustaceans, and polychaetes. Some of the larger ones even eat small fish. There are hundreds of species in the family and a number of genera, and very difficult to identify just from videos like this. So, a grenadier in the family Macrouridae.
Great, thank you, Bruce.
Yeah, thank you, Bruce. So right now you're looking pretty much the direction we're going to go, which is upslope, kind of along this crest. You saw that nice little swing showing how sharp the crest is, and we are hoping to eventually find a little denser community than we have so far today. Dave, our pilot, has looked to the west, and he's looked to the east, and he's looked to the south, and he's looked to the north, and I don't know. I think they're off screen to the north. That's where they're hiding, if they're hiding here.
Yes, well, distributions in the deep sea can be patchy, so sometimes you'll come across a couple of areas like this where you don't see anything, and then all of a sudden
there's actually inside the lens.
There's a whole bunch of stuff all at once, kind of like the buses at any number of the towns that I've lived in.
I've noticed that there's a lot of loose rock, like talus or scree in this area. I wonder if this is a fairly high disturbance habitat that gets cleaned. We've also seen a lot of dead sponge stalks, like there's something that's killed some of the sponges. Admittedly, those are very sparse here as well, but I'm wondering if disturbance somehow has an effect on the lack of life that we're seeing.
It certainly could, Bruce. So we just have no idea what the seamount has been subjected to during its very long history, which is probably on the order of 60 to 70 million years. After it was first constructed, it may very likely have been exposed to major earthquakes and other things. Presumably, it actually formed closer to the equator, to the southeast of its present location. And it's been moving steadily but extremely slowly to the northwest on the Pacific tectonic plate. So we have seen some of these kind of ridges and seamounts that have a lot of talus on there, very close to their summits, which is always very curious.
Oh,
there's a fish going off left, lower left. There he is.
Some fish.
Oh, I'm going to pull you, though.
I wonder if...
There's something funny. Has he got something in his mouth?
I can't get a good look at this, unfortunately. It's a different type of fish than we've seen before. I thought I saw a kind of a stubby head and rounded snout. I'm wondering it's either one of the cusk eels, but it could also be a snailfish, which we see very, very rarely on these dives. I think we've only seen 2 snailfish in the 2.5 years or almost 3 years of these dives. It's too bad we couldn't have gotten a closer look.
It's not over yet.
Yeah, thanks, Bruce. I think we're waiting for the ship to catch up a little bit.
Yes.
That is
and
correct.
We're just looking for things to zoom as it does so.
east in the next move.
Something up ahead.
Yeah,
I think it's
that.
dead. Dead sponge.
Yeah, looks like a dead regadrella.
Right. Some of the sediment that we've disturbed, maybe what you're seeing.
Looks like there's some kind of structure up on that ridge ahead of us. Of course, that could just be a bunch of sediment too. Yeah,
Okay, co-pilot, the ship is about to come to stop.
Another hallucination or an optical illusion.
Yeah, we need to
In
move
my case, it's a fantasy,
over the east side a little bit.
Of corals and sponges. But
I mean, we're
not. Go ahead, Scott.
Yeah, from me, it's 020.
It looks like the same on my sonar.
Yeah, okay.
So
let's go that way.
Seeing sort of a mixed substrate of a lot of rubble and boulders, and then we have these consolidated patches, which presumably is got basalt underneath it that's crusted over.
Sounds like I have a longer swing time than expected.
RV nav bridge with complete.
Manganese.
Copy bridge. Do you guys want to just keep moving?
We do want to keep moving. We just need to change our direction a little bit.
I'm looking at 020 now. Looks promising on sonar.
Yeah. 30 meters, 020.
Copy.
This is the bridge.
Let's keep moving. The next move is gonna be range 30 meters, bearing 020, speed 0.2.
That's range 30, bearing 020, speed 0.2.
Good copy.
Going up.
Maybe the substrate is just too crumbly or something too. I don't know.
Can we get a quick zoom on that little round bit on the right side? Maybe that's something that's not a sponge stock.
You're talking about the...
There's a little white circle right in dead center.
Yep. Oh, and there's a shrimp, believe it or not.
RV nav bridge, move initiated.
Okay to zoom.
Copy.
I think you spotted something there.
Is it a...
What is it?
Huh.
Can't tell if it's alive or dead.
Oops, wait a
It's
second.
like it's a test of a forearm or something.
Oh, I see.
Yeah.
So what were you saying, Matt? You saw something? I see.
Yeah, we're good. We can move on. Thanks.
Okay, why, please?
Look like a little bit like a scarab beetle.
Seems like the current has slacked off a little bit. I'm just going to float for a moment. See which way I go. Yes, much less current than an hour ago.
Looks like there's a high rock there on the upper left.
Yeah, I see it.
Oh, this terrain has got both of the Chris's speechless, and that's hard to do. We're just straining our eyes trying to see something up ahead that might be interesting to look at. And maybe we can just pass through this area as quickly as we can, and there might be something up on the summit, although the summit's not that far away. But it's about as barren as it gets. I mean, usually there's always something, you know. I mean, there has been something here.
Sure. Well, it is curious how we keep on seeing those same sponge stocks with brittle stars on them.
And there's
nothing
no,
else.
Yeah, and no real live ones either.
Yeah.
Yeah, maybe one stock sponge that could have been alive, I think.
Is that an enemy there coming to the bottom center of the screen?
And another dead sponge stock with brittle stars on it.
Not sure. Maybe a holothorium behind an old sponge stock.
Right at the bottom there.
Yeah. Or it could be an optical illusion.
It looks like fuzz to me. But no.
Well, so sea cucumber there. Nice, another transparent, probably Hansenothuria. Looks like it's depositing some sediment into the sediment pool there. That's the complicated version for saying sea cucumber poop. Oh, okay, there's something up ahead. Looks like a big stalk.
It wouldn't be it's
a sponge
a
stock.
dead sponge stock, would it?
Oh dear.
Yeah, well, sorry folks, we were hoping to treat you to another really amazing dive like yesterday, but apparently
It's definitely angular blocks there.
Apparently we couldn't come up with two in a row.
Alright, co-pilot, we are coming to a stop shortly.
Let me get lined up here. Yep. Take a sighting here.
That is weird, isn't it? I mean, not that they're, but they're all headless and that they're all brittle star, covered by brittle stars.
After it comes to a stop, Nav, give it 2 minutes to let it get a little bit out, and then you
can call RV Nav bridge, move complete.
I've got a name
Copy
for the
bridge.
Seamount.
Oh,
yeah, 345 from me.
Sleepy Hollow Seamount.
Oh, yeah. That's
Come on, this is
scarecrow. Yeah, sure. White. I mean, there's lots of Ichabod crane sponges, as it were.
Yeah. Plus the fact it's a little sleepy today too, isn't it?
Is that a what's that white thing on the rock to the right there? There's a dark
snap zoom.
Yeah.
That is.
Oh, it's a sediment.
Dead
thing.
stock.
No,
is
I
it
think
sediment? No,
yeah, it's a little
sediment.
pocket with
sand
sediment.
in a hole of sand.
Oh boy.
Why, please?
You know,
looks like there's something
how
on that
we interpret these
third
sediment
stock.
patches, does that say something
the
about our
left.
psychology?
Okay.
Oh,
Yeah, see it.
Yeah, I think that. I think that's a sponge-based map. Yeah.
Oh, wait.
There's something
what
white
is
down
that?
there.
Yes, but zoom, please. Look what I
Dum
found.
dum dum. Dave wins the Starfish Award.
Okay, landing, rolling. Hang on here. We're miles away yet.
Miles away.
Also, I think I saw a branched coral.
So last night I was looking
right
up at the
possibly
sky
a cup
and
coral
I saw a bunch
just
of
above
falling
the camera
stars.
on the black rock.
And today on our day of need, what comes in to rescue us but this orange sea star.
Oh dear, here we go.
I know, you're never going to hear the end of it.
Soon.
Was it sponges? No. It was dead sponges with brittle stars on it.
I can tell you, I think I know why we have headless sponges, and that's probably one of the culprits down there.
Oh.
Probably not him, but his
You're
buddies.
just... And looks like a goniasterid. Looks like Caliaster or something. Yeah. So this is, you're right. It's these are often observed feeding on sponges or mostly cnidarians though. This one is more, to me anyway, more of a predator on corals. But it's possible they feed on any number of things. This might be something left over from whatever used to be here before. There were lots of some of those tall sponge stalks, but or maybe it's transiting like we are. Maybe it's simply moving through this area to get to the next big sort of feeding event.
Come wide.
They're very slow moving, but you know, when you're sort of slow and steady, maybe you don't always need to
camera right.
be bringing in
small
a really big food item all the time. So
coral.
That was my starfish diatribe of the week. That sounds great. Let's move on, and maybe this is an indicator that we're seeing corals just ahead.
Are we ready for another
There's
move?
a bifurcating thing over on the horizon there. Maybe that's something.
And then we'll get one.
Bifurcating thing.
Yeah, there's like a V-shaped branch right there
Last
on
I
the
heard
top of
from
that rock.
pilot, he thought it was 0. No, never mind.
Yeah, bottom
Yeah,
right side of the
it.
screen.
That was about 3, 4, 5, but we'll jump up and look again. Here, after
Copy.
I finish this. Okay to zoom.
Yeah, I'm pretty sure it's a primnoid, or maybe even,
here we go. There's a
bridge, deck A.
That's where that sea star is headed.
It's the bridge, go ahead.
Oh, and there's a stock
The bridge, we are all set up for hot work, and with your
even better.
permission, we will proceed.
Those love
you may
bamboo
proceed.
corals.
Thank you, Bridge.
Thank you.
Yeah, and there's a black coral off in the background as well.
Or in the foreground, you mean?
No, the back.
Well, it's right here to the left.
Yeah, that's the back. Behind the
Move
rocks.
center up.
Coming right. Or do I need to
That's
come
left.
left? I was
Yeah,
distracted.
behind the rock.
Yeah, center up on main coral.
Oh, I see what you're saying. It's sticking up above the rock.
sorry,
I get it.
Oh,
I thought
yeah,
it was
yeah.
attached on the
Oh,
rock.
no,
Okay.
yeah.
Got it.
It's a crinoid. Yeah.
Wow, so this black coral. Scott, you still on? Can you give a hand with this black Antipitherian coral by any chance?
Go.
Yeah,
Go see a little
Scott
worm
France
on it.
offers the name Alternapathes.
Let's go for a
on the
two-shot.
on the forum.
yeah, thank you, Scott. Alternatopathes.
Video's clear.
Okay, wide, please.
Alternatopathes or alternapathes?
Up here.
Alternapathes.
Alternapathes, okay.
Yes.
Been pronouncing that wrong for a while.
So that tall stalk in the back looks like a sponge stalk, but it has something else on it. Maybe it's just a dead... Could be. Huh. It is still kind of kicking around, isn't it? Oh, that's kind of an odd-looking sponge, isn't it?
Yeah, what is going on here? 'Cause
Okay, Chris, go.
Clear the zone.
Go to the top.
I think it's the remnants of the head.
Yes.
Poking sideways because it did a 90-degree angle for some bizarre reason.
Huh.
Could that be what's left of a bolosoma?
I think you're right, pilot.
Does something big eat these things?
Or something with 5 arms.
But
I don't know. I think it's still... Those starfish are more coral polyp type things. I think this whole dead sponge thing is more some kind of environmental. I mean, the water currents or something.
I would...
But I mean, it looks like it's been bitten by something with a jaws of a,
Huh. I don't know. Where do you see?
I don't see teeth marks. I'm just speculating because so much of it is,
Well, so much of it is missing, and there might have been enough live
look at the
tissue
lower left
after
lobe.
the head got lobbed off to kind of start to regrow a bit. And that's why you're seeing what you see, but that's pretty weird. So I think this is actually could be what we call
Bridge, deck A.
Bolosomini new genus. It's got a nice little crinoid there at the bottom of it.
This is the bridge. Go ahead.
At that a moment
We
ago.
are finished with our hot work
And
on the O2
I'm
deck.
just saying
Okay,
that
great. Thank you very much.
just
Thank
based
you,
on the
Bridge.
color, the Bolosomini tends to have the yellowish color, but Bolosoma does
go over
too
to
in
the
some
stalk.
cases. And these little protuberances on it too, the texture of it looks like it could be that undescribed genus. These are stocked hexactinellid or glass sponges. This particular one is in the family Euplectellidae.
And we'll go full screen.
Okay.
Yeah, video's clear.
Okay.
So what makes this interesting? What makes this area so interesting? Is it the height, the sort of raised little platform?
Yeah, you expect to get good current flow over these topographic highs, and this is, well, these seamounts are pretty strange in the first place, and there are a series of bumps all over the place. When Mashkor Malik and his group mapped them last night, there is a live
white at the bottom of the screen.
Okay, so things are picking up.
Keep going.
Kind of slowly, but I do see some things off
It's
up ahead.
Yeah. There's less rubble up there too, so maybe it's a better neighborhood.
It's on the other side of the railroad tracks.
Following
I
around,
guess.
pilot.
Or the Holothurian tracks, I guess.
Oh! Another dead sponge stock with brittle stars on it.
So...
What's that weird thing on the side there? Is that another sponge?
Down low?
Dead?
Yeah, just under.
Snap?
I think it is.
Yeah, it has that strange kind of shape as the taller one.
Can you zoom in on that, pilot, right on the
I
face by the brittle star?
can.
And I think you're going to find something interesting.
Come in. I'm just floating here.
Not that interesting. I thought it might have commensal anemones in it, but okay, the brittle star is interesting.
It is.
You know, I thought it was going to be this weird relationship.
Oh no, I
between
figured that's what you were setting up for, and I was like, I don't see them. Oh well.
How do you feel about 330 pilot? Maybe a 20 meter.
Yeah, I kind of lost the picture. If that feels good to you, I'm happy.
Let's go ahead and do a 20 meter at 330 nav.
Copy.
Oh, it's like a. Oh wait, it's a sand patch. Is it a sea anemone or a sand patch in the back there?
Range 20 meters, bearing 330.
Oh, it's a sea anemone. Good
It
eyes,
is a sea
Chris.
anemone.
Way to go.
Branch coral, center
Oh, you
screen.
got the
That is
bullseye.
range 20, bearing 330.
large
Speed
black
0
coral.
This
in the
is similar to that
Copy.
one we saw the other day, that sort of yellowish one with the orange mouth and tentacles.
We're getting help on these big sea anemones, at least opinions regarding who they could be from a couple people, Michelle
Something
Rodriguez
just to the
and
left of it.
Nadia
Very large
Samarian.
arms.
Yeah, you can zoom
And
in.
right now we're just calling, we think these are in the family Actinostolidae, and that's it. That's all we can do.
Nav bridge.
As I mentioned before, anemones are really difficult.
Copy.
It's got a crinoid right next to it, but anemones are very difficult to identify from video because you got to see their innards. And unlike your beasts, they aren't transparent, or some of these big ones aren't.
Pretty far off. Let's come wide. There's some other stuff closer. Over here, let's zoom that coral that you had called out to me.
Some of those sponges are getting kind of weird looking.
Yeah, so Scott, I know you're in the middle in the kitchen with pots and pans in your hand, or you're out by the barbecue, but this looks like a bamboo coral. So if you
Just
can
come wide, I'll
rush
get a
to
toe
your
down.
home ECC and take a look at this, you'll see a bamboo coral. And it's not a double stalk sponge right in the background. I mean, we're going to look at the coral first, but is it? It's just...
Yes,
Not sure.
it is.
What's floating in the water there?
Is that a
I wonder.
comb jelly above
floater.
us? Oh no, it's a
larvacean house.
Larvacean house, I think. So there are animals that are little tadpole-shaped things, and they actually will create sort of these mucus houses that they use to feed. That's what one of those was.
Okay, zoom coral.
Okay, Scott, here we go. We're looking at bamboo coral now, so stand by.
Come on in some more. Yeah. Get twisted around here.
Yes, indeed. Like a pretty small colony. And as we zoom in, I'm going to make, I guess you're already there, but I was going to guess this maybe is looking like an isidella, but if that's the case, it would branch at the nodes, and as we're getting closer, that does not appear to be the case.
Yeah, I don't think so, Scott. I think it's internodal, as you're just describing.
And it looks like it has a little barnacle living on it as well.
Yeah. Looks like one branch, a couple of branches there gave up the ghost, no tissue on them. They didn't extend any further. The polyps are nicely spaced. You see they're not terribly dense. I do believe that I can see some prominent needle-like sclerites in them, although they're not protruding very far. The polyp size is large relative to the size of the axis, and that may just be a factor of its age. So that's not necessarily going to tell us anything about the taxonomy, but there is one clade, the S clade, which has a very small skeleton, even at a very large size. But I think I'm going to leave this as a bit of a question mark. I can't say right now what clade this
Let's go full screen.
Okay.
without going back and comparing it to some other imagery. So thanks for the close-up.
Yeah, thank you, Scott. And we were chuckling a little bit in the background there initially because we saw your chat room message that we pulled you away from your dinner and it was burning while you were on the
Move
phone.
complete.
Sorry about that.
Copy, Bridge. Thank you.
Actually, I'm not sorry about that.
No.
I'm glad you called in.
Chris, when you guys look at that in post, it looked like there were several.
What time did you call in that
something
move?
growing
Yeah.
off.
Yeah.
23:19.
Right, I saw some tubular
I haven't started moving
hydrozoans on there, and there might have been some tuberms too. Thank you. So we've got two Calophacus sponges here, and this is kind of nice, and this is also making me change my ID that I was talking with Pilot about, that really warped one with the 90-degree turn.
Sure.
Calophacus has a 90-degree turn.
Ah,
in this
yeah.
lock, and
It
we're
looks like
seeing-
it's...
It
yeah,
was
that
yellowish,
one.
though.
Yeah,
that's what threw us.
There's a slight yellow morph. This one, actually. this particular species, and I'll bet that's what we saw, this beautiful double colony, I mean 2 separate colonies shot. It's really
Let's
magnificent.
get the full screen.
Okay.
We are calling this, I think, Calophacus new subspecies something or other. I've got to look it up. Beautiful glass sponge.
Video's clear.
And it's a
Come out
stock
just
glass
about
sponge.
halfway.
And this is not in the Euplectellid family. It's in the family Rosellidae.
I was about to say, I didn't realize they had changed that.
Yeah. Okay, we don't call it new subgenus or anything. We don't even know what subgenus it's in, so we just call it Calophacus species. The Calophacus sponges are in need of revision.
Thanks, Rory.
Because we have a lot of subgenera.
Okay.
Normally you have a species, a genus, and a
Black
species
coral
like
on the right.
Calophacus maai or something like this. But with the
I think I just
Calophacus,
started
you have a
something.
Calophacus subgenus, maybe Calodiscus or some other weird thing, and then you go to species. But
looks like we're coming up on another black coral.
yeah, Bathopathes.
Did your little heel just get a little taller?
Well, I've been bouncing up and down. I pop up, get a look ahead, and then come back down. Trying to stay lined up with the axis of the ridge here. Andy, you want to fly for a while?
Sure. Yeah.
Okay, let me get back up here. Let's get a look around, and we'll switch.
Another anemone
Ooh,
center
what's
screen.
this? This is a
Snap
little...
zoom.
Oh, it's a sea anemone. Okay.
I'm definitely on the move now.
Okay, wide, please, Rowan.
Thank you.
So I'm looking, yeah, almost 340 looks to be.
Looks pretty clear out there,
Looks
too.
like the upness here. Oops. Headset cord's a little tangled.
All right, I've got to go into the Ocean Network Canada Sea Scribe and correct that one entry I made earlier. This is software they were using trying to get as many animals.
Okay, Copilot should be in the middle of a 20-meter move with bearing 330.
Okay,
to
sounds
help
good.
scientists go on and
340
possibly
was
find
my
things that they saw of
estimation
interest
when I popped
later
up there.
on and get another look at it and
See
maybe
where
get some
we end
data
up here.
associated with the animal.
The ship's been stopped for
Malik, who
about
was kind of heading up
5 minutes.
the Okeanos effort
So,
to do this.
Let's go.
It's valuable. It consumes.
So you should
a
still
few
have
of us
about maybe 10 meters
during
to
the
go,
dive,
and
trying
then
to keep up with
whether
it,
we want to keep
but
moving.
I think it's very valuable, and hopefully those
I
of you
think
who use
another
it will agree.
330
when
at
you
30
start,
would do it. I think you can go ahead and
accessing
call it in.
This new database.
Have we already seen this?
Yeah, I think we have. We can move
Okay.
up. In fact, I think there's a ledge or something up there that might have some interesting stuff on it.
All right.
To the left.
Here?
Yeah, to the
Okay.
left. Thank you.
Sure.
Three
I'll go
threes.
slowly because I'm waiting on a little bit of tether.
Of course, no worries.
30
Thank you.
meters, 330
We'll
coming
get
up.
shrimp if you want.
Thank you.
Sure. Always happy to
Go
look
ahead,
at
video.
shrimp.
This is the bridge. Go ahead.
Let's get another move in. That's going to
Video, you want to zoom in?
30 meters, bearing 330.
Looks like our old friend Nematocarcinus, a regularly
Range 30
encountered
meters,
shrimp
bearing
that we see at these depths.
330 degrees,
Long-legged.
speed 0.2.
That's a good copy.
Coming up.
Max zoom.
The zoom also shows you a lot of detail as to the kinds of things that live on the substrate that we've been sort of swimming over. There's a lot of flocculent material, sort of organic particles that settle out onto the rock, as well as some of these little frond-like organisms, which we believe to be single-celled animals, or single-celled organisms, creating little sort of tree-shaped structures. So we're coming up on this very interesting ridge.
Very interesting.
Well, it's geologically heterogeneous. It's got kind of some texture. It's got a botryoidal surface texture. Oh, and there's this
true.
And there's this coral. Is it a coral whip in the middle, or is that another
it's coral.
sponge?
No, it's a coral whip. Probably a bamboo. Scott put down that pan, rushed back to the phone. We have a different type of bamboo. We're zooming in on it now.
Coming down.
I think he just finished scraping off the burn part of his dinner, and now it's gonna happen again.
Come on in, video.
Yeah. Huh.
Oh, it's one of those things.
It's bamboo. Wow, this has got some serious talons on it.
Make it stable here.
The problem now is I don't trust a cat in there with my dinner while I'm in here. Look at this one. Yeah, this, I'm going to do a search as we go back in here, because this looks like something we may have collected in 2015, and if that's the case, it means that my lab will have done the genetics on it, and so we can get you a pretty good ID. So there is something in the I1 clay that has these pretty dramatic needles and has that general branching structure, but, oh wow, and has that general, yeah, that has these needles, wow. Those are very long needles. I also see there's some bumps right along the skeleton. It's in the soft tissue. And I believe that those are what are called the metacyst batteries. Basically areas in the tissue where there's a very high density of the stinging cells.
Track the stalk.
Sure. And those would be typically defensive. So I don't know if they are effective against B-stars or against aplocophorans, but that is a possibility. And we have a hunch that that may be something that can develop in response
hold that for just a little bit.
as opposed to always being there. So that might be an indication that sometime in the recent past
there
was something harassing the coral.
There's some kind of gelatinous thing on top of this coral.
Hydromedusa or something, right?
This is a little hydrozoan jellyfish?
Yeah, so Scott, there's a little jellyfish on the tip of the broken branch to the left, but it's not one of those feeding hydrozoans. It's something else.
Yeah, I see what you're referring to. We're not quite close enough for me to make it out.
Just hold her a little bit. We'll go full screen in a second.
Okay.
So are you cool with us putting I1 clade down as a potential ID for this bamboo coral?
Yeah, I mean, it's potential, but once again, you know, if it's in that I1 clade, then it should be branching at the nodes. And this is not branching at the nodes, so we might be better off putting it in the J clade.
We should get it on the way out,
something in 2015 at 2,064 meters, which had these really tremendously long needle-like sclerites like we're seeing here.
There we go.
I think that's probably our safest bet at this point, J clade.
Okay, video's clear.
Thank you, Scott.
Great.
Great, thank you.
It's a beauty, though.
We got a little bit more tether now, watch lead. Shall we continue up?
Yes, please.
miss something?
I think it's a dead stock. We can snap it though.
This little one, you mean?
I think the one.
What is that yellow on the rock? It's a dead sponge stock?
Yeah.
Yeah,
I'm pretty sure all those are
We can do either one if you
stocks.
want to snap zoom. I saw something small here, video, I didn't know.
Whatever you're interested in.
Want to do a quick zoom just to see?
Sure.
It's probably the same thing. It is.
Yeah.
Yep. Okay.
Some pink there on the left.
I'm getting a little more hopeful. Oh, look
Go
at
ahead.
that. Okay.
Oh, a rock pen.
A rock pen, yeah. That's a small one.
Even a little pearl star.
So rock pens are sea pens that like to perch on rocks. That's why we call them rock pens. And most sea pens, well, they live in the sea. They should. If you're going to differentiate them, you ought to call one a sand pen and one a rock pen, because most sea pens live in the sediment. But then there's this odd bunch here that just said, "The heck with that. We want to be on rocks." And so that's why they call them rock pens. Dr. Gary Williams at the Cal Academy of Sciences is working on this group.
Video is clear. Laser across.
Copy.
Could be in the genus Anthoptylum. Could be another genus too.
Happy with that video?
What's that?
Are you happy with that?
Yep.
Yeah.
All right,
Yeah.
co-pilot, the ship should be
Science,
coming
thank
to a
you.
stop soon.
I think we can get another one in there.
Let me see the
330-ish, I think.
RV nav bridge move complete.
350. Let's see. What
Copy,
we got.
bridge.
330.
Actually, let me come left a little bit. Yeah. So I'm just getting towards the edge there. So, but I think 330 is at 30
this
is
the
good.
slowest increase in density I've ever seen in my life. This is like going from the suburbs to the other suburbs.
Yes.
Copy.
We
Have
kinda
you started to
bypass the
feel
main city,
that last move?
Yes.
Are you currently moving?
or we're out
Yes,
in the country
I am
somewhere. They're in the
Okay,
main city,
I'll call
which
the
is
next one in then.
possible, but
Oh.
That's gonna be 30 meters,
I mean,
bearing
there's-
330.
real estate's cheap
Correct.
here. Like if you were to settle out here, you're not going to have to compete with your neighbors. You can
Yes, the bridge.
stretch out and breathe that good deep water.
You want to do a quick zoom on this video? Have we seen
No,
this?
let's move on ahead. We've seen a couple of those
Oh, okay.
already today. Thank you.
That's range
See, that kind of
30.
owns.
And the ones behind it too here?
Speed 0.2.
Behind, oh yeah, they're the same. I think.
There's also something right here, right between the lasers coming up.
I think it's a black coral.
Oh.
Snap it. See?
Might
Sure,
be
go ahead.
worth a
Yeah,
look.
let's
Snap. I don't know if you're interested in this.
Another rock
No, it's a star or a black coral.
black coral, yeah. But it's not the same as the one that we've been seeing. Heteropathies, maybe?
RV nav bridge move initiated.
I don't think it's
I don't know if they want a real
Piranapathies.
stable shot on this one. I will hold here.
Some type of a
Let's get the top section.
hydrozoan right at the tip.
Top
He's
section, okay.
facing the other
Get
side.
something else growing on it.
Right up here?
Kind of see him there. See him?
Ah, I see.
Soon.
Yep.
Go ahead.
Yeah, so this is a
Let's go full
black
screen.
coral,
Go ahead.
what they believe is heteropathes, and it has an interesting hydroid, I think, that is growing on one side of it. Yeah, if video is good, we can move on.
Video's clear.
Great. Whenever you're ready then. Thank you. Lasers sweeping across it.
Yep.
And here we go. What is that up there?
What's that white bun-shaped thing? Doesn't
I'll
look
find out for you.
base-like.
There's a Chrysogorgia there to the upper right of
It's
it.
a very small squat lobster between the cracks
See
as
a
well.
single polyp sclerotinian, or
Let's go
kinda
ahead
big
and
for one.
zoom in here for a second just to see.
And it is a
Well, that's a dead sponge.
Dead sponge.
Okay.
Okay, come on out video.
Thank
Upper
you.
left squat
Where did
lobster.
you say the squat lobster was? Upper left.
Zoom.
Fingers just came into.
Oh, I see.
Oh, squat lobster in the crack.
And what did you want to see after this? Watch these.
There's a chrysogorgiid coral to the right. I don't know if that's similar to ones
Sorry.
that
Top
I've seen
green.
before. There's a white thing. Excuse me. See this white thing?
Oh, I
at
see
the
it right
upper?
above it.
Yeah.
Do you want to? Are you done here with video?
Yeah.
Okay.
We don't need to see the. Yeah.
Yeah, I was going to that.
Great. Oh, here you go. We got two. I assume you want this guy on the rock, right?
That is a retracted
Yep.
sea anemone.
I'm all over the place right now. Here you go. Come in.
So the tentacles have retracted into the center of it, and then when it comes out, they'll all come out as everybody most usually recognizes sea anemones. But you're seeing mostly the base. Most of the animal has gone inside.
Video's clear when silence is.
Great. Science, are you
Oh,
clear?
good.
Yeah, we're good.
Thank you.
Come on out. Do another little hop forward here. Did you want to see this?
Snap zoom on it.
Okay, go ahead, video.
I don't know if that's the same one that you saw earlier, Chris, or there's some squat lobsters on it. I think Annie was expressing some interest in getting a little more video of these things.
Yeah, it's the same Chrysogorgia try something, I guess.
Let's see. Looks like Tina just starting to wake up
Slight
again.
tilt up.
Cystopathes or hedopathes on that black coral. Okay.
We can move on. Thank you.
You sing the message from Tina about that last one? There's a sea cucumber.
Yeah, we've seen this cucumber a couple of times, so we can continue on. Oh, looks like some sponges off to the right. Oh, good. Living sponges. Living sponges.
Now, this is a bolosoma, whereas the other ones were Calophacus. So, and how do I know? Because it's how the stalk inserts onto the head of the sponge.
Fish bottom left.
So, in
Bottom left.
Tilt
this
down.
one is inserting right underneath it, right on the bottom of it.
Oh, fish. Oh,
It's gone.
Is it a fish?
Yeah, there was one.
Oh.
It might have been. It looked briefly like a grenadier.
Back to the sponge.
You know, it's interesting. This one and the one behind it have a head, quote unquote, but there are two of them that do not.
Go ahead, video.
So it doesn't. I don't know if it's the same event. Interesting.
So this is what's called Bolosoma new genus. It's an undescribed, and I was sort of talking about how the stalk inserts onto the head of the sponge, which is very
Tilt down to
helpful
the
in knowing
stalk
what family
connection.
it's in. This
Right here?
happens to be in the family, right there. It happens to be in the family Euplectellidae. It inserts right into the bottom convex part of the head of the sponge.
ROV nav,
And so
bridge,
that's
move
characteristic
complete.
of the Euplectellid stalk sponges.
Copy,
and
bridge.
the Bolosoma, or the
Holding shot.
Bolosoma, as opposed to that other
Co-pilot,
stalk sponge
the
you
ship
saw
just stopped
earlier,
moving.
which you saw has a 90-degree
Okay,
angle.
we'll let him
and
catch
it's
up
got
a little bit,
difference
and
in-
we'll
and
get
a
another
different
one going.
location on it.
Copy.
You want me to tilt down
Kinda
with
like
it, video?
Bamboo,
Okay.
or excuse me, coral folks, where you have to know what to look for in your animals. They look for how many turns on a chrysogorgid and little
Video's
needles
clear.
in the polyps.
Come
And
on out.
sponge people look at how things insert onto other things, how it attaches to the substrate. The details that you need to pay attention to. And that's sort of the same sponge, a slightly different shape. You can get sort of morphological plasticity a bit in these sponges.
Something tall upper left.
I see it.
A few things on that boulder up ahead. Maybe dead stuff, but
looks like we have another bamboo coral here, albeit a dead one.
there's something on it.
No, there's some
These guys might
something
jump.
pink
We'll
and
get to
branching
the top.
off to the right.
Let's go ahead and go to the top.
Okay, go ahead.
Oh, wow, it's a corallium. Good eye. If we can visit that afterward, that would be great.
Okay, I think we're clear.
Okay.
You see that pilot to the right of the boulder?
Over.
As you zoom out
Pink
and
off
there,
screen left.
to the right of the boulder. Again, I don't know if it's possible, but
You got tether.
We can get there.
yeah, thank you. It's the first one of these we've seen all day.
I'll get a real stable shot then. Let's see here.
There, see, there's the Caulophacus sponge in the back. Show the difference in the way it attaches.
Oh,
See
yeah,
the 90
yeah.
degree angle. That's
Yeah.
what I was talking about.
This would be the back side of it.
This,
Bubble
this
gum.
is a paragorgid or a corallium. We'll know in a second. It's got some animalcule on the right side of the branches. Could be a shrimp or something, a spot lobster.
Okay, come on in video.
Go bottom right.
Bottom right.
You mean the
Oh.
crinoid? They look so much alike.
No, they're. You're right. It was.
No, that's fine. I understand. It was like a blob or something. Yeah.
No, I thought I saw red and waving arms. Okay, so what do we got here? The polyps are all closed. It is a corallium. And I think I know which one this is. And this is hemicorallium for sure. This is a deepwater member of the precious coral family, Coralliidae. And they harvest in some places of the world this type of coral to make jewelry because the stem you see is very hard. Thankfully that doesn't occur here because this colony itself could be 100 years old, and so it's it's
Go on full screen.
I'm not sure it's a great idea to make jewelry out of something that's lived for so long. This could be a type of hemicorallium called hemicorallium abyssal.
That little stick on the upper left might
Do
be
you want
of
to look at the
interest.
crinoid or?
Now we can move on. I think it's the same one we've been seeing.
Science, you see that little stick upper left of this? Is that a
Yeah.
Sure. I wonder if
You
that's
see it
a
a little.
Deppa sponge.
Looks like a little Q-tip. Yeah.
Did we want to go behind this? Or
No, there was a little sort of like a thin stick right where we were.
Okay.
I think it's the Q-tip thing.
Right there.
That's
Okay.
it.
Yeah.
And why would we be interested in a little white stick? Because it could be a really cool type of sponge, but I'm not sure it is.
I think it is. We're just seeing it on the side.
An amphipod?
I'm not sure. I think we're seeing it on the side. That might, what Chris is thinking is this might be a carnivorous sponge, a cladorhizid, but those usually kind of look, the ones that we've seen before have kind of a central round disc with spines coming off of it, and it looks like it might actually be, but we're seeing it from the side angle.
Oh, there you go. Yeah.
So yeah, these can apparently adhere, or what they can capture prey by using those spines, which adhere to the spines, and then they're eventually digested later on. Some of them actually will create sort of a fake cellular stomach, and then they'll send cells down there to digest them because they don't have a full
Yeah, if you
digestive
could
tissue
call
type
in
system.
320 at 30.
Carnivorous sponges are weird things.
Shrimp, top right. May not be of interest, but
Any interest in the shrimp watch leads?
The shrimp.
top of screen.
Sure.
Okay, that's
Go ahead,
30
Ian.
meters, 320 coming up.
Thank you.
I'll let him just drift in here.
He's motoring by. This is a swimming shrimp.
He's not going to drift in anymore.
He is now attacking.
The bridge.
Going away from us.
We're ready for the
It's
next
retreating.
hold. That's going to
Is
be
he retreating?
range
Oh, okay.
30 meters, bearing 320, speed 0.7.
That's
yeah,
range
sure
30, bearing 320.
Did you know that those kinds of shrimp were jealous of other shrimp, Chris?
Uh-oh. No, I didn't know that, Chris.
It's because they have Peneus envy. I guess you didn't get it. Peneus? Like the genus?
Yeah, okay.
Oh.
We're
okay,
gonna get in
nice.
trouble.
Jokes.
Want to see this?
It's vertebrate zoology
Yeah,
humor.
please,
Go ahead, zoom in.
that thing.
I'll be here all week.
Yeah, there's also a sponge on the lower left that is different.
ROV nav bridge.
When you zoom out, you'll see it.
Okay. Let's see.
It's on the side of there. It is that yellow thing in the bottom of the boulder.
Oh, over there.
Yeah.
Let's see. Go ahead, video.
Oh, that's a weird looking
yeah,
thing.
So this could be my buddy that we collected yesterday, this yellow Poliopogon species. And it's in a very weird location where it is, but you see you just don't get members of this genus with this yellow color to it. And it looks like it was damaged. You see this upper
Yeah.
right here.
I'm starting to
Very
wonder if
strange.
there's just something here that's taking a bite out of these things. Maybe it's that grenadier.
Well, you don't see these sponges that are predated on very often. They've got some serious defenses in the form
What
of
about
all
that
their
sponge?
spicules. What's that?
Oh,
Which
looks
one?
like there's a cloud of rise on the right.
In the focus here.
Yeah, this looks like a, well, if this is a cloud-arised sponge, then conceivably those thin
Should get moving here,
projections
video,
off to
whenever
the
we're
side are what they use to capture small crustaceans.
Actually, did
or
we
other
get that
food.
small one? Do you want to zoom back in one more time?
No, we got it.
Okay.
Oh, that's fine.
I'm good with it.
Yeah.
I'm looking 320, and that's where we're headed.
Headed to 320.
So things are looking up, even though we've seen that sea cucumber before. There's more of them. We're seeing them more frequently. So that may indicate that there's more food, and thus more productivity, and thus more animals.
Something's right over here.
Some nice pillow flows here. These big sort of bulbous
I know,
rocks.
so lovely. Sometimes it feels like I could just be jumping along those right now.
Is that an anemone at the base of that sponge?
Yeah,
Oh,
it
no,
is.
it's on the rock. There
Yeah.
it is. Another one of these big old things that we call Actinostolidae.
Here's a spiral something on the right side there.
All right. Do
Yeah.
you want to see this up on the? Okay. Where do we want to see first?
Let's look at the coral first. Sort of the one that's
You mean the anemone or the coral?
no, there's a spiral coral on the
Oh.
right side there.
On the right side?
Yeah.
Well, how about I Edge of snap the zoom on screen. the anemone since it's here?
Okay,
Oh,
let's do a
sorry.
snap zoom.
Yeah, yeah. Sorry to sound back and forth.
Yep. You just
Yeah, I don't know if this is a characteristic, but you see how there's those lineations on the disc. Those were the same lineations we saw in that small one that was withdrawn. So I think you're right. It's the same thing.
Yeah, and we did think that a few years ago until somebody straightened us out. We were actually calling these anemones
Exosalactus.
Wow.
And then we got a different
Video's
opinion
Come on,
and convinced us that it wasn't.
So they said there was a coral?
Yeah, to the right. There's a spiral. Sort of.
Oh, back in there.
Yeah.
Okay, sure. Let me go back there. So we'll just pass on these guys right now.
Mashkor, what is the matter with your Seamount? There's not very many animals on this thing. What's the story? Yesterday you did so well on that other Seamount. Yeah, we did. Look at it. It's all rocky right here. Yeah, well, we're getting there. But, you know, I don't know. I mean, Mashkor Malik has joined us in the control room for a few minutes, and he is the person who runs the team that did the marvelous mapping of the Seamount last night and early this morning.
Try to get to the tip here.
So you mentioned what this is, right, Chris?
This is some kind of octocoral, I think.
Yeah,
but
it's Iridogorgia.
It is Iridogorgia. It looks really beaten down for.
Yeah, it's not great, is it? It's a little
yeah, I didn't
ratty
recognize
looking.
it. I guess I could mean that there's a starfish nearby.
Hello, co-pilot.
Good afternoon.
Just to let you know, you are currently in the middle of a move, 30-meter move at 320.
320, copy that. And the ship is still moving?
We should be coming to stop soon.
Soon, and we'll go for another 4 or 5 minutes then?
Copy that.
Copy that. Thank you. Also,
RV nav bridge move
previous
complete.
co-pilot wasn't sure if there was something in...
Copy bridge. Go ahead.
He wasn't sure if there was something in the port rock box.
But a bamboo.
So there is both
any interest
rock boxes are full.
Let's take a peek at this.
We have something in the port inner bio box, which might float out. So
okay, so don't open the port at all.
Your dinner should
Copy that.
be totally burnt
And
by
how
now,
about
or you
starboard is
consumed
empty?
That is correct. No biosamples on starboard bio boxes.
Copy that. Thank you.
That's got these extraordinarily long needles.
Yeah, I'm having a look at it.
Okay.
This may be what we were imaging earlier, and what's really informative is that one that we saw earlier did appear to have branches, and this one is not. And so this raises one of, as you know, my favorite things to talk about, and that is the difficulty of discerning these species
slowly to the base.
just based on their colony growth form, because you don't always know what stage of life you're seeing it, and whether what you're looking at is a branching form or an unbranched form. And the way the current taxonomy is set up, anything that doesn't have a branch and would be in the subfamily of bamboo corals would be put in the genus Lepidices. But as you've heard less than I say
pilot, we are
times,
looking down and maybe a little back at you. So
taxa, and we know that
soon.
There's lots of different ways to build a colony, and there are other things you have to look at besides simply the way they branch.
Yeah, thank you, Scott. And I think you left it at J clade. Is that correct? Or I think you were toying with S clade or I1 clade. I'm trying to remember now.
Yeah, sometimes I never know either. I did leave it at J clade before, and I'm not entirely satisfied with that, but I think that is the way we should leave it for the moment.
Okay. Thank you, Scott.
In fact, I will go further to say that in my database, I'm looking at images from 2015 when we collected one of these J-clade unbranched whips with these very, very prominent spines, and it looks a lot like what we were just looking at, and it's why I hesitated before, because it was a branched one. So nice to see the unbranched one.
Hey, pilot, I'm going to turn my head to keep you in view.
Copy.
You need to come back to
coming
port
up on a
when
nice
you get
glass
underway
bunch.
again.
Well, let's take a look because there's something on it. This is one we've seen earlier today, but it might have some commensals on it. Right on those white things.
Oh, yeah. Yeah.
Okay, come on in video.
Snails? Yeah.
Oh, that's an interesting one.
Wow.
Huh. Interesting.
Tilt down a little bit.
Slightly.
Maybe I've seen snails on these sponges before, but I can't remember.
That might be too far.
Are they hermit crabs?
No, there's the.
Oh, that's
the
a
two
hermit.
tentacles.
No, that's a hermit crab.
Are you sure?
Yeah, see the leg coming out on the side.
I don't know. Those look like tentacles, and then those look like the eyes of a snail. Well, at the very least, I mean, like, does that one looks like a snail, right? Because I think they're attached with the proboscis. I mean, that one might be a hermit crab, but the other ones look. Let's see. Are there any others in view? Maybe we're missing something.
You want to look around for other ones?
Yeah.
You said? Okay. Zoom out
Well, there's
slightly.
one to the
There's
left.
one up top here.
Yeah, up top here.
You want to see that one
just get
too?
a little better look at it. Well, I think that's the snail. That doesn't look like a hermit crab to me.
Yeah. Well, so at the very least, there are snails here.
There's one on the top left there. Do you want to see that one?
Doesn't seem like it shows any better.
We'll try video, see if I can do a stable shot here. With tilting up.
Smack zoom.
Copy.
Was that Caulophacus?
Something red on the top right. Something red at the top?
Sure. So sponges, especially glass sponges, are what one calls ecosystem engineers. Their physical structure provides habitat for different types of animals.
Blown out wide.
We've often seen a multitude of different animal phyla present on different glass sponges at depth, and we can see a perfect example of that here. This one has, for example, 1, 2, 3, 4, 5 small snails living right on the surface. But if we were to turn it around or look around at some of the other glass sponges, we'd probably see brittle stars. Sometimes little crustaceans live in there. I'd almost bet that we'll probably find some if we were to look through it. But sponges are an important component of any ecosystem because they literally are an ecosystem for a bunch of other smaller animals. How's that?
Lasers, copy that.
Okay, we can move on. Thank you.
You're wide. Your mic is not open.
Watch this. We're just going to get quick lasers on here and switch out pilots.
Lovely. Thank you.
Roger that. Thank you.
Is our light out here on the panel? Yep.
So we're having a bit of a pilot change in the front row here, so stand by. It'll just be a minute.
Welcome aboard, pilots.
Howdy, howdy.
Howdy. I don't know if Andy told you our heading was going to be 320, so I turned to keep him in view, so you need to go to port and push out. Yeah.
All right, copy that. Sorry, what was that, Chris?
Oh, nothing. Just saying, how's it going, guys?
Yeah, it's going great.
Good.
Where is everything?
Oh, well, that's a good question.
It's
that last time I was piloting, I mean, you couldn't even sit down.
There's stuff over that way, whichever that way is.
I'm assuming we've seen this glass sponge.
Yes, sir.
All right.
I think moving upwards and seeing what's on these higher platforms is of interest.
Roger that.
Thank you.
Ooh, our motor installation's way down. What's going on? Now it's back up. Strange. Can you note that in the log?
Yep.
All right, Don, if you can pull up a little bit.
Okay.
I'm pretty close to you right now. Yeah, it's down to 7,700 km.
Josh, I don't know if they relayed to you, but in general they wanted to make tracks.
Yeah, copy that.
There's this dark...
Yeah, come on in video.
Blob. Whoa, what is that? It's some kind of slug.
It's a pleurobrink. But
It is a
very
pleurobrink.
unusual one.
Whoa, it's 10 centimeters across.
It's going to the edge.
Chris, do you know which one this is?
No, and actually I would offer this as a possible collection option here because these are really rare and this is so different than any I've ever seen. And it's large enough, I think we have a chance.
Wow.
Pilot, what do you think about this? Is there any chance we could collect this?
We can give it a shot.
I'll throw it out to the group here.
Let's get some good imagery first.
If you are getting caught up with us right now, we have a really strange pleurobrink. But
Pilot, what was that number that you saw
Chris and I
earlier
have never seen
on
it
the
before, but
insulation
maybe
resistance?
one of you has.
So the motor insulation resistance is
was 17,
dropping down.
Yeah, 13.6.
Yeah, periodically.
Yeah, but yes from Scott France.
Which is a bit concerning. Wow, that thing can really move.
And he's about to drop off.
The color also suggested yes. Yeah, we got a consensus. Everybody is giving a very
There was
enthusiastic
something
yes.
else up on that rock to the left as well.
Yeah.
It's dropping again, 77.
There's a red
Yeah,
sort
I
of
saw
circular
a red
thing.
something or other. But
Yeah.
whoa, that is different. Holy moly, yeah. What is that red thing?
Look how big it is.
Yeah.
It's like nothing, 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 and then boom.
He's going to head over
giant
that
monster
ledge pretty
Cthulhu
soon.
pleurobrink.
All right, what do we have in bio boxes?
We've got something in the left bio box. We prefer not to open that one.
All right, copy that.
So.
All right, can we ramp up hydraulics?
Copy that.
Yeah, port inner is the only one, but it's a floater.
All right, so we're going to go starboard.
Yes.
This is amazing.
Nothing in starboard other than the rock box. Nothing in starboard by.
All right, positioning is going to be important on this one. Let's see. Let's do a zoom on him. Let's get some good imagery before I go in.
So while we're sort of looking at it, wow, that thing is nutty.
Actually, let's
gorgeous.
come out because he's going to crawl under.
He's going to
that
crawl
rock.
under. That's what I was worried
Yeah.
about.
I was going to say, what's that red thing on the rock to the left? But
Yeah, let's get this guy in the box and we'll look at that
Yeah.
red thing.
All right, let's get that box ready.
Copy that.
So pretty rare to see sea slugs down here.
Yeah,
not
I
a
know,
common
it's really
animal at all.
strange.
Well, any sea slug, even the
Yeah.
other more familiar
Yeah, I mean, I thought that they all pretty much just pinch out after, I don't know, about 1,000 meters or so.
Can I help you with anything?
We're good for right now. Don, if you want to bring up
Mini Zeus.
Yeah.
Copy that.
Wow. That's experience for you.
Grab mid-water. Nice catch.
Kids, don't try this at home. Actually, they could if they could build a manipulator arm and try it on little kids like these. Come on.
Now he's out of reach. Alright, I gotta reposition and go for him again. He's not escaped yet.
I'm going to tip down on Mini Zeus.
I don't need it.
Nope. Okay.
Just, let's open the drawer.
All right. Okay. Coming out. It's all the way.
All right, now you can tip down on Mini Zeus if you want.
Okay, drawer closed.
He's a little damaged, but he's in there.
You got him. Thanks very much.
Wow,
for
thank you.
Great collection.
Hey, video.
We saw another deep sea.
Is there any way you're able to pump the
hydrophone
hydrophone through my headset alone?
Earlier on in the expedition, a few days ago, Terry Gossliner gave me a name.
Thanks. If not, don't worry.
Wait a second. Pilot, before we leave here, there was a red thing on the opposite side of the little boulder there once you stowed the arm that I wonder if we could swing around and look at.
Yeah, absolutely.
Thank you. And good job.
Thank you. Whew. All right.
Hydraulics off.
All right, I'm done
that's
with,
copy. Is there any way we can record the sound
with
somewhere else right now?
Camera.
Nope. Okay.
There he is, the red thing.
Thanks. Can we record this video?
Come on in video.
You say come in or
Yes,
live?
come on in.
What is that? Oh, it's an Anthomastus. It's closed up.
So you got something?
Yeah,
Is he eating something?
I've seen them many times.
That's noteworthy. It might, maybe that's what the side gill slug, what the slug was eating. This is actually a good sign as far as the region, though, because if there's a slug like that moving around, maybe it means there's food for it to eat. So we're probably, I bet we're just above a huge field of things.
I hope you're right, but it's definitely getting better. It's pretty dry in the beginning, but we're definitely seeing some corals and sponges, live ones.
Compile, did that go in starboard inner?
Yes. You got the waypoint?
Copy that. Yeah, I got it down.
Oh, there might be a
survey to nav.
Something to the left in the water column. I thought it might be a fish. Oh, shrimp maybe. What is
Hey,
that?
Roland says you can record this hydrophone thing that I'm saying right now.
Oh, Ketognath. Okay.
Oh, that's great. Thank you so much.
Not used to them being so rigorous.
Let's get the next 5 minutes.
Oh wow!
At least.
Nice shot of.
And can you get anything previous or no? Okay, from here on for 5 minutes, please. Thanks.
Wow. Sort of see the teeth on the side and everything. These are one of those animals that if they were as big as we were, we would be terrified of them.
Inside your lens,
they can
minimum
see
focus.
in a fairly wide range around them, and they have giant teeth that can swallow animals that are the size of their own body.
Do we want to get a ship move going?
Their Caulophacus sponge.
You talking to me, Don, or are you talking
We can,
both
yeah,
of us,
if you guys are ready.
I thought the idea was to cover some ground.
Oh, we've been covering some good ground. We've been covering some good ground, Don.
Okay, copy that.
Let
There's
me, I
an
can show the whole
open
thing.
one of those we just saw on the
There's
left.
another Anthomastus just to show you that this is the open one. The other one was closed.
Go ahead and roll.
Do a look at this. It's these are mushroom corals. So it's got probably on the order of somewhere between 15 plus individual polyps that are coming out of a ball-shaped structure. Is it? Let's see where.
Oh, I think I see what Roland's talking about.
But,
that's one of the actual.
So if we
arms
average
of the polyp
0.08 for
putting
the
food
rest
into
of the dive, we can get to the very
I think
end.
he's talking about. You're talking about that sort of top polyp with the red.
So I can put a move in as soon as you guys are ready for one.
That's actually it's actually caught something and it's sort of moving something into the. Into the
You looking
mouth.
at 320? You happy with that?
320 looks good. Yes.
Copy that. I'm going to give you a 30-meter move. Bridge, RV nav.
It's the bridge. Go ahead.
I'd like to put in a ship move, please.
Okay, go ahead.
Range 30 meters, bearing 320 degrees, 0.2 knots.
Range 30 meters,
There's a lot of lava
bearing
coming
320
down from
degrees,
this peak at some
speed
point
0
millions of years ago.
Good copy.
Coming up.
Let's take a look at this little white circle to the left.
You're gonna have to
It's
help
on the
me,
rock.
Chris. All right, come on in.
Weird.
Maxon.
Sponge?
ROV nav, move initiated.
Copy.
Are those polyps? I don't know what we're looking at. Oh.
Yeah.
I wonder if it's...
Could it be a sea slug?
Well, yeah. I was wondering if those are the gills coming off of it. A weird...
Wow, that'd be weird to find two sea slugs at
I'm doing that.
2,300 meters all of a sudden.
Pull out for you.
Yeah, I don't know.
So there's a snail. Is that a snail there down there to the left of it, or to the left?
Or is it just a dead stock that we're looking at?
Yeah, probably a dead stock. We can move on. But there's a snail down to the left there. Oh, you know what?
This is the bridge. Go ahead.
Hey,
Gaza.
quick question for you. Is that bow thruster moving around pretty good right now?
This little shiny iridescent
just a
snail
second ago?
we're looking at right there.
It seems about normal. I don't see it right now.
Gaza's,
I wasn't paying any attention just a few minutes ago, but
in 2015
well,
are bigger than
spinning
that.
up
Well,
that
no, if
horizon,
it could be a small.
if it
much
drops
pointing off
and
our
rolls,
starboard
then
quarter.
Copy that. Thank you.
No, I'm serious. That's apparently their behavior.
Right, come on in.
Yeah, I know.
Yeah, I bet that's what it's about to do. Yeah, that's sponge stock.
Yeah.
Well, we could wait all day for that to drop and roll, but
I'm not so sure it's Gaza at all. I think we're fine, pilot.
All right, come on up.
They actually measured the distance they were rolling for Mary Wickstein, who wrote that up, published the observation on it, and I looked a lot at that video footage. So if it is one, it's a really tiny one. Yes, they have shiny shells like that, some of them, but
Just a small white dot at the top. Seems to be a stock something or
Come
another.
on in, Roland.
oh, it's a nice amphiurid brittle star.
those little forams, arborescent forams, a little cluster of them, I guess. At least that's what we think they are.
Yeah.
All right, coming up.
It's very hard consolidated substrate here.
Oh, there's a fair amount of stuff living around it, though. It's not so bad.
It's quite a crack right in the middle of it as well.
Yeah.
All right, let's come out.
That's another one of these anemones that's partially open, at least. I wonder if we snap zoom that straight down.
Straight down right here.
I think it is.
Come on in, Roland.
Unless it's another dead. It's another. Never mind. Thank you. Wishful thinking.
Oh, sponge stocks. They're just.
It's a sponge base or coral base or something weird.
Yeah.
Oh, look at the tether. Got a loop in it.
Oh, that's a dead sponge stock.
We got a loop in it?
Yep. Check out the Titan.
Tilt down on your Zeus.
Yep. There it is.
All right. So.
Well, maybe it's not so bad if you just.
Well, can you zoom in on it? I need to do a port turn. So.
Tip. Video, can you zoom on
no,
the
you can leave it. It's fine. I can see it.
Okay.
It's underneath. All right.
Yeah.
So pull up a little, Don.
Coming up.
And I'm gonna get rid of this turn. Just a second here, Chris.
No, I can see already in the co-pilot screen. No problem.
Go ahead, survey.
Josh caught a twist happening in the tether before it happened, so he's.
Yeah, could you give me maybe 2 more minutes?
Of a maneuver here
Thanks.
to make sure they don't get a twist in the tether. So he will be with you momentarily.
You tilt down on your Zeus.
Coming down. Going up a little bit more. all the back
we're
I've
getting
got.
the high-level view of what's ahead, and I'm not sure we're going to get into that incredible community today.
ROV nav, move complete.
Copy, bridge.
So now you're showing 1.7 turns. Huh?
So let's just look at the tether here.
Yep, I'm going to stretch it out. You want me to come down a little bit?
That is quite a coating of manganese. Look at that to the left all over the substrate.
Yeah.
I think I may have connected a lot of loose boulders there, or maybe these are funny, well, they're lava flows, I guess.
They come down a little so we got some more
Manganese
light.
crusted lava flows.
Looks good.
Looks good.
Yeah.
All right.
Okay.
Looks like we came down with a turn. Good spot down.
Thanks.
So we're at 2,200 meters right now.
This is a long shot, but there's a white ball right in the middle of that lower screen crack. Can we just take a quick zoom?
You're going to have to direct me a little bit here.
So there's a dark angular corner in the lower
you see
center.
the lasers relative to the lasers?
Oh,
down left.
Down, and then to the left.
Straight under lasers.
Yeah, right there.
Come on in.
And then more to the right. Yeah, right there. Some kind of... Oh, yeah, it's a sponge stock or something. Okay, thank you.
There's a purple there. What's the purple?
Oh, what's that? Is it sort of a pteropod shell? Oh, it doesn't matter. Yeah, that's fine. Thanks.
Okay, moving along.
Is it flattening out, or we still got some more climbing to do?
It's starting to flatten out. At least in high pack.
Looks like there's something on that sponge surface.
Let's take a quick look at this sponge.
Come on in.
Hey, Chris.
Yeah, oh, sorry.
Oh, no, it's fine.
Oh, look at this. Wow.
This is your thing, isn't it?
Yeah, but these are different. Look at the sizes of these things. These are commensal anemones that are growing out of some kind of sponge. I'm sorry, I was addressing the the chat room there because there's a discussion on how to preserve that sea slug, and we've got a bunch of people who had some recommendations. But I've got to turn my attention here to this. Whoa. It's a hard sponge. It's can't really tell if it's broken or what's going on with the top there. And then all these yellow parts are a zoanthid, looks like a zoanthid or hydrozoan, I guess, that are probably commensal on the sponge. Wow, that's a lot. And you can see some of their bodies on the other side. I can't tell if this is a tube sponge or what, or whether it's a plate. I don't suppose you could swing around and look at the other side, could you, after you're done with this?
Yeah.
This is a very different kind of commensal anemone that we've seen before. This could be something we call Tretopleura. There's also maybe a Heteropathes black coral.
Go ahead, survey.
To the left of it.
Okay, we're good. Yes.
Do you want a partial zoom as you turn?
You can just hold this for right now. Did Fernando ever get someone from Team Electrical?
Well, if the sponge is paddle-shaped, then
For
most
what?
likely it's in
about the motor?
family Incinitaridae and in the genus Tretopleura.
We're going to call around to a warehouse?
We're defaulting to
Oh,
on all of these
to workshop?
paddle-shaped sponges
they
for
should
now.
be in the hangar.
Yeah, come on in.
They're not known to have commensal
Workshop,
anemones,
Nav.
which is weird about this.
Hey, yeah, we're looking for somebody from electrical. Could
Those are
you
just
come
big
upstairs, please?
Weird, isn't it?
Commensals, yeah. those
No,
are really
just
big, the
somebody from
biggest I've ever
electrical,
seen
Team
with
Electrical.
it.
Yep. Go to the control room,
There's
please.
not as many on this side, maybe, or
You can just tell him what the problem is.
What's the problem?
The motor DC, the motor insulation resistance is fluctuating.
Okay. Hey, workshop, Nav.
He's barely on his way up. Doesn't look like a tube.
No, it certainly doesn't, does it? So I'm going to say Uncinitaridae or a Eurydidae, one of the two, but and then with, I guess, colonial anemones or zoanthids or something.
Something.
Wow, that's a very fascinating new observation. Thank you very much for that view.
Go workshop.
Oh,
the motor.
I
I
saw
don't know how to plot it.
small things on the bottom there.
You might want to come up and show us how.
At the base of that sponge we were just looking at, there's a whole bunch of small, tiny sponges and other things.
Did you want to look at those?
Yeah, I wasn't sure if we had moved on, but yeah, if we could maybe, as long as it's still in our range.
From that front view, it looked like there was
come
some
on in, Roland.
carnivorous guys.
Yeah, workshop.
While you're doing that, I need to finish answering this chat here about the slug preservation.
Copy. Yeah, just send somebody up.
Yeah, so there's a combination of different things here.
Just send somebody up, please.
So
to the control room.
obviously there's this black coral, and then there's these thistle-like things, which have the sort of ladder-like projections across
Do
them.
you know how to
it's got a nice brill
plot
star
them?
living off
I have
of it.
zero idea.
Me either, so.
Or maybe it's something caught on a carnivorous sponge. That's an interesting idea. Yeah,
It's maxed out.
It's hard to say. But, interesting. And then I think farther, if you pan out further, there's more of these little round, sort of, I guess they're baby Caulophacus or something. I saw a patch of them, I think, on that same rock surface towards the end of the... Yeah, they're to the right there.
Yeah, come on in, Roland.
Yeah, see all these sort of little cute cute little sponge babies. You know, there's like 4 of them in one little patch. Hey, Chris, this might be something you might actually want to look at.
What's that?
Oh no, all these small sponges, and they're all in the same area. I mean, sort of a kind of a reproductive settlement cluster or something.
Yeah, it certainly could be. It's pretty cool.
Anyway,
little babies.
Yeah. Anyway, we can move on. Thanks.
Copy that. Thank you.
So the reason I was silent here is that we've got some recommendations regarding the sea slug preservation, and some various people were weighing in on this. Scott France got us connected to James Aubrey, as well as Megan McCullough, and also somebody, let's see.
Well, they're talking about Angel Valdez, who's one of the biologists. I think he's at CSU San Luis Obispo. He works on sea slugs.
Yeah, for some reason I thought I saw a message from him, but maybe it wasn't.
No, I don't think he's.
Scott was copying. Yeah. Okay. Oh yeah, from Angel Valdez, right?
Yeah.
So, does everybody agree that you'd like a piece of the foot and ethanol for the 95% sequencing? Because it's not too difficult, and then we can put the rest of the animal in formalin. We do have enough. Is that are we going to follow? Is that what people would like? What's the consensus here? Because I've never collected one of these animals before, and I certainly don't want to mess this one up.
Should we get a move underway now?
We'll wait for a moment to get a response. Meanwhile,
fingers crossed.
Take a look
USPL's
what's going on the sea floor here.
Say again.
Might have
run out of.
So yes,
there's
you
a
want
lot of these
you're looking at 326. You still happy with
I get
that?
the impression this used to be a fairly
or
active area.
Yeah, 320.
320, yeah.
Okay, 30 meters.
Is there a waypoint we're heading towards?
Yeah, it's right up here, but we're just trying to get
up on that
higher.
plateau. Yeah. Okay. Yep.
So, okay, I'll get you a 30-meter move at 320. Bridge, RV Nav.
Scott's saying it might be relaxed.
It's the bridge. Go ahead.
Hey, can we get a ship move, please?
Unless I hear otherwise,
30 meters at 320, 0.2 knots.
Unless I hear
Range,
otherwise,
30 meters, bearing 320.
What we're going to try to do is remove a piece of the muscle of the
Good
foot.
copy. Thank
Okay,
you.
now we got a response. Yes, we could chill this down for a bit as well. We do have a freezer aboard, and we can certainly try to chill the animal down to see if we can relax it before preserving it. So how about this? We try to chill it and try to get it nice and cold, and then we will take a piece out of the foot and put it in 95% alcohol. We're also going to take a small piece and put it in the OGL fix.
Video swap it out
for their
what am I looking
Ocean
at?
Up there on
Legacy.
the, is that another?
Are you talking about
and
the tether?
Then the rest
tether,
of it
yes.
we're going
Sorry.
to put in formaldehyde, or not formaldehyde,
guess it's
10%
just
formalin. Excuse me.
How about you keep your heading here where it is?
If
and
that's
I'll push
not
ahead.
acceptable, let me know, because that'll be our
Sorry,
plan.
I didn't know what you were talking about.
Yeah, yeah. Sorry.
I thought you were pointing at the winch, which
No. Yeah.
got me very concerned.
Megan says, can we slowly introduce ethanol to reduce the contraction? We haven't so far.
I guess that's all right.
That could be a little bit difficult, but we could try it. What do you think, Matt? Are you seeing this? Because we could trade off on this. I'm trying to think how we would do this. So I'm assuming she's suggesting we have like a put it in 10% ethanol first, and then 20, 30, etc., and try to do it that way. Okay, so do we have a tentative method here? How we're going to do this? This will. Yeah, what we're going to do, it comes up, and then we can try to photograph it at least initially as quickly as we can, but on the other hand, we could try to put it in the freezer really quick, because the photographing it is just going to warm it up. That's my only concern. But we can try to get it in the freezer and chill it down, but not freeze it. Right. Yeah, well, I guess it's a matter of whether it's fully contracted or not. If it's contracted up, then we can try that. But then we will remove a piece of the foot in ethanol, remove a very small piece of the foot also to go in OGL fix, and then the rest of it goes into formalin. And in the ethanol, we're going to need to do some changes so we'll mix up some dilutions for the ethanol. Yeah, because that's a small piece, if you see what I mean. So we're going to have to make a bunch of dilutions of the ethanol for that. That's it. Well, that's what they're saying, that introducing the ethanol slowly will do better. No, so we're doing various things. The bulk of the animal is going to go in formalin. It's just straight up in formalin.
Oh, pilot,
you're
can
right.
we take a look at
You're
this
right.
whip in front of us? Thank you.
Absolutely. So Don, if you want to use the Titan, try to keep an eye on the tether here.
Yeah, okay, copy.
All right, you're right. I'm getting mixed up here. So you're right.
All right, come on in, video.
So after the formalin fix, then yeah, we'll definitely do the...
It's a partial.
Yeah.
So it looks like we have some kind of bamboo coral here. And we can usually tell that because the skeleton that is below all of this tissue is white with dark nodes, and that gives it the superficial appearance of bamboo. Two of our scientists on shore, Les Watling and Scott France, have done a lot of work with these animals, and maybe they can help us out with which one this might be. It seems to have these very long spines coming up from the polyps. And Scott France declares this to be part of his jade clade of bamboo corals. This is an allusion, or this references a molecular tree that he has, which designates each of these clusters with different letters. So probably
I can give us a few minutes to get another ship move in there. You guys are happy with that? That one just finished.
Thank you, pilot. We can move on.
All right, video. When you're good.
All right. We're wide.
All right.
Can we take a quick look at some of those tracks down in the mud?
Where are you looking?
Center, bottom.
Right there?
Yeah, any one of those would be fine.
All right.
So Oftentimes we see sea cucumbers or snails or other kinds of organisms, and they live amongst the sediment here, and they leave tracks that stay here sometimes for quite a long time. Sometimes you can see the animals that make them. Yeah, I think this is fine. I thought maybe we might be able to spot whatever was making the trails.
All right.
Thank you.
Let's come on out.
Okay, pilots, we have about 30 minutes left on bottom.
Copy that.
Well, actually, I don't know if you heard, we have 30 minutes left.
Roger that, 30 minutes left. Thank you.
Let's take a look at this.
Yeah, the red coral and the sponge both, I guess.
Yes.
Right, come on in, video.
This business going on this rock. Oh, there's quite a lot of it going on.
Oh, this is a interesting coral. It's sponge too. Both.
Come and take. I'm going to approach it, so
Okay.
you'll have to adjust focus.
Okay. So this is Hemicorallium, the coral. And the sponge is something that we call Ferrioneroca erecta because it is a form of a species that we have identified tentatively as Ferrioneroca, but it's an erect form, which means it doesn't form a bush. It forms kind of a columnar type of colony. And we haven't seen very many of these so far on the cruise. I'm pretty sure that's what it is. It differs from those leafy sponges that we saw yesterday. And what's that red, bright red thing on the top there? I don't know if you can get a tighter
Some kind of interesting
yeah.
little...
Yeah, come out a little bit.
Yep. How's that?
Yeah, hold that. I'm going to sit down here, Chris, to get a nice steady shot of that.
Sure, thank you.
Just take a moment. That rock is...
I did too, so but I'll just go ahead and do it anyway. And I'm keeping time, so then it was Hemicorallium, I'll get that one.
Right,
we want to average 0.1 to get to the end of this waypoint.
Copy that.
Huh. Yeah, I can't tell from that even. Nothing, there's no details to it. Maybe an amphipod. Or maybe an ostracod, I guess. Not sure. Oh. It's always nice to see brittle stars close up. They have such interesting structural surfaces. This one has, this is a member of the family Ophiocanthidae, I think maybe in the genus Ophioplinthaka. It has these very distinctive folds on the disc as well as all of these small, they're either spinules or granules on the disc, and it has a number of arm spines that are quite sharp. Looks like it's crawling over this Hemicorallium. Yeah, I think we're done when video is done.
Yeah, copy that. Just pan down, see if we see anything, and then...
Yes, absolutely. Thank you.
All right. And you're good, video.
All right. If we can just get kind of a full specimen shot, that would be nice.
Yeah, you want to kill lasers down.
Oh, let's take a quick look over to the left there. I think that's another carnivorous sponge.
Oh yeah, I see it now.
Watch these. Do we want to make it up to waypoint 3, or are we okay just spotting these and getting the close-ups of these guys?
So, oh wow, look at that.
I wouldn't mind getting up to waypoint 3 if we could.
So
okay,
this is another nice carnivorous sponge.
Moving.
You can see the
negative pilot. The ship has stopped.
They will hold fast small prey.
Co-pilot, are you still good with 320?
Move the items to the center of the
Still
disc.
looks good.
These are actually not possessed of true tissues.
Just
so they
0,
actually
0,
form
0
sort
look
of
any better?
unusual kind of stomach-like structures with their cells, which phagocytize, which is to say they use the cells themselves to digest food. We caught video of one of these the other day.
Yeah, we're picking up targets over that way too.
Yeah,
okay, I'd like to put that in. It's closer to the waypoint.
Thank you.
Okay.
All right, video is good.
Come on up.
Let's try that and see what it does.
Bridge,
Full
RV
light.
nav.
This is the bridge. Go ahead.
Ready for ship move.
Okay.
30 meters, bearing 000, 0.2 knots.
Range 30 meters, bearing 000 degrees, speed 0.2.
Good copy.
Get this floater.
ROV nav, move initiated.
Copy.
If you can come in tighter, go for it. He's gone.
Yeah.
There's a question by Scott France whether there'll be a post-dive call, and I'm going to go out and consult with Kelly about that, because while we can't really tell you much about tomorrow's dive site, we can tell you about the dive plan and the discussions about the dive plan if you'd like to hear that. So just give me a moment and I'll be right back.
Snap in here. Alright, coming out. Get these as you come by.
Oh, good. More of these whip corals, bamboos in the, what are they calling them, J-clade.
Right, coming out.
Oh, here's a nice glass sponge ahead of us.
Okay, wow, we're doing all types of things, getting ready to leave the bottom here. So we're trying to get some advice on preserving these two animals we collected today, because I personally have never collected either one of those type of animals before. So thank you very much, those of you who commented on that, and we are taking copious notes here to help us out. The second thing is there is a question regarding whether there'll be a post-dive call, and we don't have any bathymetry to show you for the next site because we're going to map it tonight, but we would like to have the call because we'd like to discuss with you the plan for the rest of the cruise. And Kelly Elliott is going to bring up the Google Earth image that shows you how we've sort of modified the plan a little bit and get your feedback.
Hold that.
So if you'd like to be a part of that, that's going to happen immediately after the ROV leaves bottom.
Wow. So these are the tests of forams. These are the structures, the sedimentary structures made by giant amoebas. they vary in sort of shapes and by which kinds of species that made them. I think these are in the group Xenophilophoria, but there are other groups as well. A lot of these can send sort of tentacular extensions through the sediment, and they can actually devour animals. I'm not sure if that's the case with the ones that made these structures, but these are apparently things made by single-celled animals. They're quite remarkable considering how big they are.
All right, coming out.
Thank you. It's also interesting to note that all those areas that we were just in, all the sediment's been disturbed, which suggests that there's some kind of perhaps sea cucumbers or other beasts moving through the sediment. Oh, that's an interesting crinoid. Let's take a look at that. Here. No, it's the same one that we've been seeing. It has these reproductive structures on the pinnules along the arms. Yeah, we can move on. Thank you.
Ship still moving, Nav?
Currently, yes.
Okay.
But it's not for very long. It will be coming to an end. Well,
RV nav, move complete.
Copy that right now.
So Megan has talked about tincturing seawater,
would you like me to give you a minute or
alcohol
two?
or cocaine.
Or you want me to go ahead and get that next call in?
The issue, of course, is time and shipboard and what you can do on a small lab on a ship too. So
yeah, so one more will basically
appreciate the
get us
comments.
there,
the simpler for us, the better, because we are usually busting just to keep up with all of our work. But I threw out some suggestions how we're going to deal with the slugs, and as well as the slug and the Seriantid, and hopefully that'll work reasonably well.
There's your sea cucumber.
Here's another, Let's might as well take a quick look.
Bridge, RV nav.
It's got a nice head-on view. We haven't had that perspective yet.
This is the bridge. Go ahead.
I'd like to put in the same ship move, 30 meters, 000, 0.2 knots.
That's full zoom.
Range 30 meters, bearing 000 degrees, speed 0.2.
Good copy.
Coming up.
This one's one we've seen before. I believe it's similar to Hansenothuria, but I'm just getting a shot of this from the head-on angle because you can sort of see the 5-part symmetry that you see along the body. You can usually determine that straight away from sea stars because their bodies are usually splayed out radially. Sea cucumbers have it, but it's oriented along their body rather than sort of splayed out in a nice pentaradial pattern. Oh, there's another sea cucumber on the rock there. There'll be lots of food here
as we come along.
And the tentacular surface there, that's the mouth. Yeah, that's the mouth.
That's a funny one because it's got all of those little round ossicles or whatever they are in the walls.
Okay.
We saw this earlier, and all that Dave would say about these ones that have these spots is that he thinks they're synallactids based on, of course, the transparency and other characters.
It's a gorgeous beast.
Yeah, it sure is. Yeah, thanks, pilot. We got some good video of one of these a little earlier, too.
Yeah, copy that. Alright, Thea, when you're good with this shot.
Okay. And we're full wide.
Thank you.
So what's this feathery thing we're
Come on in.
coming up behind? Oh, it's a black coral. Okay. Probably looks like one we've seen before today.
Come in tight.
Yeah. What's wrong with this picture? So it could be Stauropathes, maybe, because it's got a couple sub-branches on it. Pretty weird Stauropathes, if that's what it is. It's definitely an Antipitherian. So one of the diagnostic characters for Stauropathes is sub-branching. Again, I don't know the exact terminology. Sub-branching, you can see in the left there, from the pinnules coming off of the main branch. Probably secondary pinnulation might be the word for it. That's characteristic of Stauropathes, and this one definitely has that. Yeah, thank you very much.
All right, come on out, video.
Is that a sediment patch in the background, or there's a rock right behind the black coral that's star-shaped.
Where are we looking, Chris?
Up a little bit over to the right, and then up about an inch.
Nope, right.
Oh, sorry. It's
just
rather
left of
small
lasers.
on my screen.
In the middle of
yeah,
the rock.
it's a sandy patch. Never mind. Thank you. We can just head on in whatever direction you were going.
Okay, Andy, the best way to direct me is relative to the lasers.
Yeah, okay.
Yeah,
because
slowly learning.
You say, oh, it's it's between those two rocks, and
yeah, I know. I'm sorry.
yeah.
I'll think. I need to think more in terms of.
Or which segment of
context.
the screen?
Yes.
Top left.
Yeah. Sorry, you'd think that after all these years of watching Okeanos, I'd be able to do this when I was in the seat. Thank you.
I mean, it's tough. It's not easy, especially on terrain like this.
It's probably another corallium. And what is that? It's a dead stock. Never mind.
Let's get the possible corallium here.
All right.
Got creatures on it.
Flyby. All right, come in tight.
More of these. What are
snake stars?
Yeah. I think Scott was calling them master of schemat, or Les was calling them master of schematids yesterday.
Mm-hmm.
Just let me know if you want anything tighter, Chris. Otherwise, I'll keep going.
I'm good on this one. It's hemicorallium. We got a pretty solid ID on it.
All right, copy that.
Okay.
Come on out, video.
Okay, pilots, you have 10 minutes left before you need to start stowing.
Copy that.
RV nav bridge, move complete.
Copy. We'll be coming probably to end of that move by the time it's time to prepare. So I don't think we need to get another move in.
There's a sponge on the top of that boulder that we haven't seen today, I think. I don't know if that's on your way, but
that's where we're heading.
You can do a snap zoom on this. This is a Caulophacus, a little bit different species, I think, than what we've seen so far today.
Video.
Yeah, this is something we call Caulophacus oxydiscus, I think. I gotta look that one up. I'm spacing out right now. Rosella stalked glass sponge, beautiful one.
Looks like it has some commensals on it. Oh, is that what is that? Shrimp, baby?
Let's get the commensal.
Yeah.
It was oxy. Okay. See, brain cell fired actually in my head, and I had it right.
Oh, is it a bentactinophore? Yeah, it's a bentactinophore.
Oh, good.
Yeah, so this is one of the more unusual groups of animals that live on the surface of these sponges. This is a ctenophore, or comb jelly. The swimming versions are probably ones that most people are more familiar with because they're really brilliantly iridescent. If you shine a light on them when they're swimming, they look very beautiful. But there are also a subgroup of these comb jellies which live on the bottoms of the seafloor and on substrates. They fish, in a sense, with long tentacles by extending their feeding tentacles into the water. And it looks like it's just exploiting the water current that the sponge is doing in order to sort of find food. I'm not sure I'm seeing the tentacles, though.
It could be retracted.
Yeah.
Thank you, pilot.
Okay, come on out.
Or full wide.
These are pretty wild when these things get old and huge. They can get very, very large, as we found on one dive site in 2015. They can be enormous.
Well, just the ones yesterday were enough to...
Yeah, so on this boulder, pilot, if you pan up just a hair, you'll see one of these sponges that we may have seen yesterday. This one on the right-hand side. Sure looks like it's the same type of sponge that were all over the place yesterday.
Yeah, I remember those were those weird axe-shaped things.
Right, come on in.
Yeah, so this is in the family Phariidae. And this isn't that Pharia oca. This is probably in a different genus, probably Aspidoscopula, I think, is a better guess for this one. But the family is fairly certain. And a beautiful hard glass sponge, very different texture than the one we just imaged, that stock sponge. Even though they're both related, they're both glass sponges. This one has essentially glued all of its spicules together to form a very hard structure, whereas the other one, that ball on top, is all soft and squishy. It hasn't glued its spicules together. So,
just to the left
very
of the
good.
stock.
Log this one. Seeing a lot of new stuff right here at the end.
Oh, there's another little amphipod or something living inside it, or on it.
These things are beautiful, if you ask me.
Something living inside it, yeah.
They're just gorgeous animals.
Oh,
very fragile, but...
Oh yeah, could stare endlessly through the lattice.
Yeah, very thin-walled sponge.
Yeah, it's a shrimp.
And a lot of the Phariids and the members of that family, oh yeah, there's a shrimp in there. A lot of the members of that family are very thin-walled.
Josh, did I hear you guys say there was 10 minutes left?
There is about 5 minutes left now.
All right, come on up. Did you copy that, Chris?
I'm sorry, I didn't. What was
that?
It?
5 minutes?
5 minutes.
5 minutes. So now is probably a good time to just do a nice recap. This is Chris Mott from the Smithsonian's National Museum of Natural History and Chris Kelly from the University of Hawaii. We've been surveying this region, this unexplored and previously unmapped Seamount, and it's been filled with a lot of potential. Several glass sponges. We've seen rock pens, some corals such as Chrysogorgia. We've seen a number of different black corals as well as some sea anemones. We saw a really neat polychelid lobster, Homerion asper, early on in the dive, one sea star, several sea cucumbers that looked like they might have been in the family Synallactidae, a fair number of fish, some grenadiers and a halosaur, but the big brown cherry on top of this sundae was the plurobranchiid sea slug. At least I think that's the group it belonged to, which was easily 10 centimeters long and not something that one typically finds at this depth. Most sea slugs tend to occur in much shallower water. So that was collected, and a number of specialists will be working hard to figure out which one it was. We actually saw another pleurobranchiid, I think, last week, but which was not collected, but still very interesting. Chris, do you have anything that you think I missed?
No, I think you did a pretty good job. Just sort of noting here, we're very close, if not on the summit right now, and we got a fair amount of sediment on it, which is a bit surprising. Usually these are pretty current swept, but still pillow lavas. We're seeing the very top of this seamount. So anyway, every site is different. We've said this a gazillion times, and it just every day we do proves that correct, or proves it to be right. Please hang on if you would like us, if you'd like to listen to the post-dive call. We are going to discuss the rest of this cruise with those folks who want to discuss that with us, and let you know what's going on, and it's going to happen in just a few minutes. So in the meantime, enjoy these last images of Sleepy Hollow Seamount.
Yeah, come on.
Oh, we're calling that now?
Yeah, Pycnogonid.
Oh,
where is it? Oh, look
right
at
at
this.
the end.
Right at the end.
So this is actually kind of cool because this is a sea spider, a Pycnogonid, probably in the genus Colosendeus or Colosende. These are actually predators on cnidarians, and if we zoom in, I bet we'll see that its proboscis is doing something that people who like this coral won't like. Yeah. I think it might. Notice that all the polyps are gone.
Where's his end? Like what end is end?
Yeah, it's the one facing. It's in direct contact with the stalk. So these are predators on cnidarians. There's a study that they did in Monterey Bay that shows them sucking juices out of sea anemones, and we've seen them multiple times sucking juices out of other different kinds of cnidarians. I think so. In this case, what I think it's doing is, you notice again the polyps are all missing or withdrawn, and the proboscis is just sort of attacking it one polyp at a time.
Excuse me, pilot. I'll just throw this out. We still have a few minutes. I don't suppose there's any chance you could grab a snip off that branch. Could be one of the very few occasions that we could actually collect one of these pycnogonids.
Yeah, we can try that. We'd have to go into one of the bio boxes. You said there was a floater for it.
Yeah, but the slug will probably be okay down in the bottom there. I guess we could take a look.
Okay. Yeah, we can try it. Do we have time now?
We have 30 seconds.
All right. Come wide, video.
Okay.
All right, still the swing arms down. Let's bring up the...
Copy that.
As I'm doing this sample.
I think without hardly any current at all, you could try the port side. Those tentacles will probably stay put if you take the box out slowly.
I mean, we could try the starboard side.
Okay. You think this is a good idea, Chris, to try to grab one of these, or are they pretty well known? Or?
Oh yeah. Oh no. I mean, at the very least, the catching it in association with the prey item is a valuable reason for it. I'm just merely concerned about the time, so that's fine. I would suggest it. I endorse it.
All right, so let's try for this.
Today was the day of odd collections.
Okay, and we're thinking starboard bio, correct?
Well, quality over quantity.
Well, yeah, we like to get a variety of different things.
Make sure you come outboard with that lower one first.
I am.
Yep. I just saw it.
Copy that.
I think you might be behind it. Yeah. It's very narrow.
All right, hold on the drawer.
Copy that.
Okay, go ahead on the draw.
Okay, extending. I'm going to tip down on the mini Zeus for you.
I get the arm in an awkward position.
You want me to move mini Zeus? No, the uppers are still out. I don't want to do it because I can't see them yet.
Okay.
Lowers are stowed. You want me to move Zeus, mini Zeus rather?
You can leave it where it is.
Okay.
Yeah, you can move mini Zeus, please.
Okay, copy that.
I got my arm all twisted up here.
How's that?
Did the draw come out anymore, or is that it?
I believe that's it. I'll try it again.
No, I'm having so much trouble with this.
That's all.
Alright, there we go.
A little more light help if I bring that upper arm in and down.
Well, it went in with the sea slug. Alright.
Okay.
Let's close it.
You're clear. Draw, retract.
Alright.
Great, thank you very much.
Sorry about that, I went in with the sea slug.
It's not a problem. I think we're going to be able to keep them straight.
Alright, let's get you lined up.
That's okay. Yeah, that's okay because it'd be actually pretty confusing. He meant to go that way. If they were both the same bio boss, they would assume it was a mistake. But yeah, we're good. Oh, pretty interesting way to finish this dive up. So in literally seconds, we're going to go ahead and move into post-dive call mode. So hang tight. I don't know if Kelly Elliott is going to come down there and guide this one instead of Mashkor. I see her getting up through the curtains, I think. Somebody is.
Okay. Some reason I'm not able. Okay, that's there we go. That's why.
Hi guys, are you still there?
Yes, we are. We're waiting for Kelly to bring up Google Earth. We just need a few minutes to get set up to show you the maps.
No problem. I just got back, so I wasn't sure where we were.
Okay,
yeah, Scott, if you just got back, we just collected a Picnic on an Irida Gorgia as a final act of the dive.
Yeah, I saw that. So I've been able to see the video. I just can't hear anything, and so
we'll come up with a positive turn here.
Yeah, and thanks to your comments, Asako, too, for
you want me to get on
a little bit of a slow dive, but it finished with a bang.
Yep.
So that was good.
Ahead.
Okay.
All right, hi folks, this is Kelly here. If you are tuned into feed 3, you should now see a map showing Google Earth display.
Bridge, RV nav.
This is the bridge, go ahead.
We are coming off bottom.
Thank you.
There you are. Okay, we ready to start climbing?
Just stand by.
Stand.
Alright, we can start coming up.
Okay.
We want to note, Nav, that we have a positive turn in.
Okay.
Up to full speed.
Come up to speed. Coming up.
How far off are we on time?
Let me check. 3 minutes.
No, it's not bad at all. So, what happened there is I grabbed it in a way that made it very hard to stow.
I saw it when you went to stow it. I was like, whoop.
Yep.
So
might have.
I didn't think about it enough
Yeah.
before I grabbed it, and it made it really hard to stow it in that bio box because I was rushing. So don't rush. Smooth is fast. Fast is slow.
I cleaned up some of the USBL.
How'd you do it?
Changed the threshold.
The threshold?
Yeah, on the transceiver itself, because like this is the ping from D2, and it's getting super washed out. And so I turned up the threshold, and it appears to be working right now, but we need to do a test.
So
we'll
you
see.
turned up the threshold?
I did.
What does that mean?
I increased it from 15 to 19. There's not a lot in the manual to talk about it, but...
Okay, so it's not. So it's if you increase the threshold, that means it's not going to mistake other noise for a ping.
Well, that's what I'm not sure about. That's what I think is happening.
Okay. We'll have to check the manual.
Yeah, and I was reading the manual and it was not very clear, so it's going to be something I talk to the manufacturer about.
All right, we are off bottom. No more bottom lock.
Excellent.
Nice job there at the end, Don. It's a bit of a rush. They always,
yeah.
For some reason,
I
Chris
know.
always
Right
likes to
minute.
ask for a sample, like when we have 30 seconds left.
Right.
It's like he's trying to challenge me.
Yeah. So with this positive turn in, they're going to need to run the rope.
Yeah.
Which way? Let's see. If it's a positive turn. So they need to hand it over and under. Is that right?
I don't know. I need a. The only way I can do it is
a piece of string,
piece of
two
string.
pieces.